Howdy, and welcome back to another episode of World Bigfoot Radio.
When we first began the SASQUATCH Genome project and we ended up publishing our paper, we found that the peer reviewers weren't really concerned with our science. They seemed to say that our science was okay. There was no critique to it. What there was critique of was they just couldn't believe the results, and even stated so in some cases. They actually said that it just had to be contamination, and that was the main complaint, and people that haven't read the paper have just picked up and run with this statement. However, we have graphical proof that they're not contaminated, the samples aren't, and I'm making a video to that effect. However, I thought before I released that video I would let y'all enjoy a little clip out of France. Dr. Tom Gilbert, who's a geneticist at the University of Copenhagen, very famous man. He's published over 125 peer-reviewed papers. He's done a lot of work on ancient human origins, and is one of the top people in the field. And he actually sequenced some hairs from Sumatra, from the orang pendek. And his state-of-the-art famous university laboratory is not going to contaminate the samples. However, in this clip, not only does he say that his results were human for mitochondrial DNA, just like ours were, but he also is followed by the hair analyst that says the hair wasn't human, just like what our results were. However, I had to laugh because the person that did the hair analysis also accuses Dr. Gilbert of contaminating the samples. This is so ludicrous, I can't even tell you. And so I thought you would enjoy seeing the clip. Don't take my word for it, watch the clip. And enjoy the video. And next video I'll try to post will be the one where I'm pulling out some of our raw data to show there's no contamination. Hope y'all enjoy. Bye for now.
And we are back with somebody that I've been wanting to have on the show for a very long time. And we finally managed to make that happen. So I'm like super excited for it. And actually I had a little thing here that I was going to pull up and let you guys see, but apparently I don't have it loaded right now. But it was a direct quote from my guest from years and years ago from morning news on, I believe it was ABC, talking about how the Sasquatch are not big monkeys running around the woods. They're actually a kind of people. And so with that shoddy introduction, let me introduce the amazing Dr. Melba Ketchum. Welcome to World Bigfoot Central.
Thank you for having me.
The pleasure is all mine. I've been a fan for years, as I told you, and I know you don't like the word fan, but I'm in this to do the actual research. I do the field research. I got a bunch of friends that do the same thing. We try and gather the evidence, and we need people on the academic side of things to sift through that evidence and try and make some sense out of it, which is why I think, every single actual person involved in the legitimate Bigfoot community owes you a huge debt of gratitude, and they should all be sending you cards, letters, and boxes of chocolate. So I'm really glad to have you here.
I appreciate that. Definitely don't need the chocolate. Once on the lips, forever on the hip. But I'll take that as a compliment. I did get a cookbook from somebody because I love to cook. Anyway, I guess it's a natural thing because of the lab. Mad scientists in the lab, mad scientists in the kitchen. It's all about measurements.
So
Anyway.
for the benefit of anybody that may not already know who you are, can you give us a brief rundown on your illustrious career that caused every troll on earth to attack you?
I graduated with a doctorate in veterinary medicine from Texas A&M University. It qualifies me to do genetics because we do have genetics in school, as well as the fact that most European laboratories that do any animal testing at all are run by veterinarians. It's not like that in the States. For whatever reason, they prefer PhDs, but it's all the same. As I became allergic to my patients, I ended up going into genetics and began doing animal testing and even human testing and forensics, as it turns out. So we did a lot of disease diagnostics and color diagnostics and all types of different things with the animal DNA, but then we went into paternities and mixed forensics, which is human and animal. A lot of times, like a murder case, they would send us hair that was found at the crime scene, and we would test it against the knowns from the suspect's house. So different things like that. We've done death penalty cases, Innocence Project cases. We did a little bit of everything. Every year we would get a few SASQUATCH samples for species identification. However, we always just got goats or raccoons or something. It was never anything interesting, and we would laugh about it. I did not believe they existed. I thought people that believed in Sasquatch were believing in some kind of fantasy. I never dreamed they were real. And so as far as other things I did, I worked on the sequence analysis of the World Trade Center victims. In fact, I recently got a commemorative coin sent to me for my work with that, and which was really touched me because it was a great thing to be able to serve my country.
I was actually about to bring that up from what I had heard when they made up the list of who's super expert that can show up here and take squished remains and figure out what it came from, that you were really high at the top of the list, basically.
Well, we did, and there were 22,000 samples that had to be analyzed. There were people that were in many parts, and it was terrible, and it was heartbreaking. And what we learned when we did that was it just would tear your guts out. But yet we were able to give closure to a lot of families because we would, analyze the remains and then, of course, match them against family members. And so it was a very humbling experience. Internationally, I was elected by my peers internationally to chair several genetics committees, horses and dogs and what have you, on the DNA end of things. And so I served like 4 terms on the horses and 1 or 2 on the dogs as chair. So, I was at the top of my game until I did Sasquatch. Seriously.
But on the other hand, if we had to, really put a wish list together and go, who could we wish for that could actually do Bigfoot DNA, Sasquatch DNA genome testing that's like really, really good at it and is going to do a really awesome job on it, you would have been on the top of that wish list too.
Well, maybe, I don't know. I didn't ask for it. It came to me. I quite did not plan on doing this at all. 1, I thought it was a waste of time. I didn't think they were real. And 2, I'm serious. But we got asked to do some death information truth samples. And we got a little bit of an interesting find on one of them that was supposed to be a Yeti, but it wasn't enough to do anything definitive at all. But whenever people found out that we tested that sample, they deluged us with SASQUATCH samples. So we had over 100 samples to test. And, you know, I found out by looking at them that the hair looked a lot alike on all of them. Now, I'm not a hair expert, but doing forensics, I knew somebody who was. So the project we just kind of backed into it at first. And we took the hair and had a hair analyst look at it. And all the unknown hairs were put in one group, and the ones that he could identify, because he had knowns for every animal in North America.
Right.
The ones he could identify, we culled those out before we ever started testing, because there were so many and the cost was so prohibitive. And it's gotten a lot cheaper now, but back then it was extremely expensive to test. So anyway, we went ahead and started testing those, and we were not the first to test the mitochondrial DNA on these things. Actually, NYU had tested some. One of Adrian Erickson's samples, the Matilda sample, they had tested, got a human sequence, and Paleo Labs in Canada had tested the same DNA, because he split the sample into several portions. And they got the same sequence as NYU, and then when we sequenced it, we got the same sequence. So that was a 3-time tested sample, and we all got the same thing, human mitochondrial DNA from that sample. So, I don't claim to be the first as far as the mito. And then the second thing that we did that nobody else had done at that point that I knew of was we went ahead and started doing nuclear testing. And we did something that would tell us if it was real human or not. We took a bunch of the samples, the best samples, and sent them in to USC and had them do what's called a 2.5 million SNP chip, which is it's where you take the human point mutations, 2.5 million of them, which is a lot of the genome, pretty much the whole genome, and you test them against the sample. And it has to be above 97% in order to be human, 100% human. I drew my own blood and degraded it, let it sit out on the desk till it stunk, to put it crudely, as a control. And then I put some regular human controls in it. We had like 4 human controls, and then sent in all these SASQUATCH samples. And true to being a hybrid, which they are, a human hybrid, they showed a pretty wide range of below 97%. None of them made it to 97%. They ran from, I think about 56% to 89%, I think was the highest one, because they are not human, but they are also not an ape. So we did that kind of as a screening thing. And this Matilda sample came in. I don't remember her without going to the paper, but she was obviously below human. And we did other tests with her DNA that showed that she actually matched her picture. She carried the red-haired gene, and she carried the gene, the skin color gene does not show up. You can do what they call universal primers, which is a way of, it's like bookends that delineate a certain sequence in your DNA. And to get that sequence, you can widen it out a little bit and create primers that'll test like all mammals. So that's what we did. They're called universal primers because it would test any mammal. And it had to do with skin color. And a lot of them have gray skin color. And Matilda had gray skin color. And so when we ran all these samples on this 2.5 million SNP chip, it came in as not a single one of them had any SNPs related to that gene. And then we ran the gene separately with universal primers, and we still did not get anything. So their sequence for their skin color is totally different than anything on the planet. and it won't work with your universal primers, which is very interesting. So she had the gray skin, and she had absolutely no reaction anywhere or no sequence anywhere for skin color. And so that right there would prove that she was not a human.
You know, that matches up perfectly with the description of Xana from over there in the Caucasus Mountains, where all the eyewitness reports say she had gray skin too.
Yeah. I don't know about her, but I'll talk about that in a second. Back to Matilda. She had the red hair. She had the skin tone and the lack of, that's where negative results actually told you something, because she should have run. And she was also female. We only had 2 or 3 females in the study out of 100-something samples. So with that said, you know, if you did it statistically, just that one skin color gene would throw it into infinity of her being a human being. She's not a human, but yet she's, you know, got human mitochondrial DNA. So it was a well worked up sample. Regrettably, we ran out of DNA on her. And as far as Xana, we have a tooth from Xana and from Quit.
I'm showing everybody the picture of the tooth right now, by the way.
Oh, okay. I'm not sure if we're going to do them or not. It'll depend on how much research money we raise. They're at the bottom of the heap because Dr. Gilbert at Copenhagen, who's a very good scientist, allegedly ran her, and the results did not turn out like they were expected to be. I would like to verify his findings. If we keep getting some donations, I would like to run those 2, because it would be huge if he truly is a Sasquatch and not a African. It would be huge for the simple reason that we have a half hybrid with Quit. And supposedly not only she had these attributes, but he had them too. He had great strength. Allegedly he could pick up a chair with somebody sitting in it with his teeth.
Yeah. Yeah, if you look at the picture of him, he's a really rugged looking dude too.
Right. But I do believe their human fraction, if they're more than human, the human fraction I do believe is African though. Because all of the children look like African Americans or what have you. They have the hair and the
wide nose.
Yeah.
Yep.
So,
actually, I don't know if you've talked to Rich Soul at all, but he's on that too with the MUC7E saliva DNA marker thing. That again is from the tiny population somewhere in sub-Saharan North Africa, and might be a potential marker for Sasquatch too.
Well, we don't know because we weren't allowed to load it up into GenBank. We did get it compared in GenBank and others, but we were not allowed to upload our results. And that's on my website, where they kept just putting us off and asking for signed consent forms from them and everything else stupid. Anything to keep us from uploading our data, which was not right. But we were able to compare it. So we do know what they are and everything.
So they still haven't uploaded your data. They're still being obstinate and unscientific, huh?
Yes, of course. It's the government. They don't want it out there. Nobody wants to go to a national park where they're being stared at by giant hairy creatures.
I don't know. I do it all the time. I think it's relaxing.
Well, you know, I think that people with little children and stuff are going to avoid it, maybe.
Yeah.
There's
ways around all that, though. I mean, they got certain areas they like to hang around, and you put up a sign, hey, Bigfoot baby season, stay out. Take it down again midsummer. There, problem solved.
You know, the problem is with all of it is that, you know, the government just doesn't want it out there. It's one of those things, they don't want it out there. And so, you know, they made sure of that when we started to publish the paper. They absolutely got to all the journals. We had to find a new journal. And apparently it was under the radar, so that's the only reason we were able to publish. We did get our peer reviews we needed, and we ended up having to buy the journal to actually get it out because they backed out within 2 hours of when it was to go live and published. The lawyers called the guy and said, do not publish this. It will ruin you. And so they backed out of publishing it. They had gotten the peer reviews and everything. We didn't. And they backed out 2 hours before it was supposed to go live. So we ended up having to buy that journal to get the peer reviews that we didn't do, and to get to be able to publish it. And that's perfectly kosher. As far as the rules, we have that on the website too. All right, back to how we tried to publish. When we got ready to publish the paper, it was obvious the government didn't want it out, because we cleared the first round at Nature. And being a peer reviewer, normally if you clear the first round and you make the changes in the paper that the peer reviewers request, you eventually get it published, whether it takes 1 or 2 or 3 rounds or whatever. We did everything they requested. And when we came back to Nature, after we passed the first round, we had one that wouldn't review it again, one that had reviewed it and passed it and said that he wasn't qualified to do this. He was the first round, but not the second round. We had one that didn't even read the paper both times because he asked for stuff that was already in the paper. And the third one, he just made fun of it. He was not serious at all. It was very unprofessional. And his final take on it was, I just don't believe it. So those are not peer reviews.
Yeah, that's not scientific. I don't believe it has nothing to do with the evidence.
I know, and that's exactly what, they're on my website. You can read the peer reviews. And the other thing they kept blaming it on was contamination. And on my YouTube channel, I have a whole video on contamination. We have proof it was not contaminated, because when you run a lot of these tests, you get a graphic representation of the sequence or whatever you're testing. And if it's like a overhead projector in school where you have a transparency that you lay on top of something, if you have 2 profiles in there, say you got a human and you got a dog in the same profile, you're going to have peaks that overlap, like you've laid one profile on top of the other. And it's real easy to tell when it's contaminated for that reason. We did not have contamination. We're used to doing forensics. You get contaminated samples all the time, or mixed samples where you have more than one contributor. We are used to dealing with that. That was not an issue at all. So, and that's why I put out that video, because everybody was saying, "Oh, it's contaminated," and that's just the only thing they could come up with to try to fault it, because there was nothing wrong with the science at all.
Mm-hmm.
So, anyway, we went to Nature first, and then they turned us down on the second round, not the first round, which means it should have published. And then the next thing that happened was I went to PLOS, because I peer reviewed for them, and they're pretty easy to get past. They'll publish stuff they really probably shouldn't. And they wouldn't even take it for peer review. And they'll take any old garbage. And so, I did a Chinese paper. I think it took us 4 rounds before they got it in enough shape that we could actually, use it. And so I know that they should. And also, I believe it was them that published a new ape in Africa that had not even 1/10 of the amount of DNA sequence that we had. And that was just on short term. That wasn't the whole genomes or any of the longer stuff. This was just little short gene sequences. They basically did mitochondrial DNA and didn't even do as much as we did. So,
but yet they declared that a known species, and they won't even look at your work.
Exactly.
Which one is this? Was it Homoliflora zeanthus or Homo luzon?
No, this was a monkey.
Oh, monkey. Not one of the teeny humanoids. Got it.
No, at the time, I don't believe they had done the genome for the Homo florensis yet. So that was not done. That was later. But regardless of it, they're different anyway. I'm sorry. They're different anyway. They're comparable to other hominids. And the thing is, Sasquatch is not.
Mm-hmm.
They have a lot of unknown DNA in them. And when you look, there's a national depository called GenBank. And what GenBank is, it's where every scientist, when they get a new sequence, they upload it to GenBank, or even if they get another of the same sequence, like say more than one human sample, they'll go ahead and upload it. And then it's used supercomputers to compare all of this. And whenever you compare it, then you say, well, it'll come back 99% human, 95% human, or what have you. It gives you the top species that it's similar to if it's different. We got nothing similar. Nothing on the paternal side.
So the nuclear DNA, there's nothing even remotely close to match it with.
Well, there is, because there's human and stuff, but there's this huge chunk of unknown that there's nothing to compare to at all. Mm-hmm. Zero. And so, even in the smaller sequences before we did the genomes, I have a letter from one of the scientists at one of the labs we outsourced to. It's a very good lab, and they're accredited. And it's a letter from one of the PhDs there that asked me, because we blasted this sequence, and she says, it's a perfect, clear sequence. She said it is, you can even see the primers on both ends. And it's so clear, and it's 100 bases shorter than it's supposed to be. And we put it in GenBank, and it came up, nothing matches it. Have you discovered a new species? I have it in writing. They ran the test. They blasted it in GenBank, which means search through GenBank, and came up with nothing. Well, this is something I really haven't released yet. I'll talk more about it, but some of the newer findings, we got the percentages of unknown, and they're large. So in the paper, we already had it with some of these others. Like, we kind of targeted the Y chromosome. We had so many males that we targeted the Y chromosome because we wanted to know who the daddies were. And we did various different tests from sequencing some of the gene, some of the amylogenin gene, which is a gene that's used in forensics for sex determination. And we also did YSTRs, which is the male equivalent of mitochondrial DNA, in that you can trace back your male lineage all the way through your ancestors through this, because all male lineages will follow down. For instance, if you have a son, your YSTRs are going to be identical to your YSTRs. Just like a female, the mitochondrial DNA is going to be the same as the mother in all of her children, but it doesn't pass down from the male.
How many generations down from the original mother does her mito DNA pass?
Well, infinitely, really.
So, but not with the males.
No, yes, with the males, it's just a different test. It's a nuclear test, and it has to do with the Y chromosome. It's got these little fragments, length fragments, called STRs, short tandem repeats. That these length fragments will identify you and all of your male offspring and all your male ancestors. So if you basically came from let's just say you came from a Middle Eastern lineage, that Middle Eastern lineage will carry all the way down, because it doesn't change really, unless there's a mutation. So you can trace back, many, many, many generations and thousands of years with it. Just, that's why they talk about the mitochondrial Eve. Well, this is basically the YSTR atom. So you can go way back, and these are some of the things we use to age these things. But anyway, they did not run well on the YSTRs. They do not have them. I only got a few hits on like one of them, and we did a whole panel of them. So they are not. It's just not human. It's not human. It's unknown. And so we we've got this unknown male contributor. So it was very interesting because we couldn't link it to anything. I mean, some people say it's Gigantopithecus. Well, for the majority, it's unknown DNA, and now they've got Gigantopithecus, and it's an orangutan, and I mean, they're all related.
Yeah.
And
I know enough about primatology to debunk that one just by looking at their foot morphology, so you can forget about that silly notion.
Yeah, well, I'm in agreement, but I'm saying the DNA will also tell you there's no orangutan in it.
Mm-hmm. Yeah, and they don't know that. Has anybody extracted Gigano DNA yet? I hadn't heard that they had.
Yeah, they have now. That's why they know it's a precursor to an orangutan.
There you go. So it's not related to Bigfoot, period.
No, not Gigantopithecus.
Nope.
And so, but they are genetically engineered, and there is some weirdness in there that I haven't released, but I will be releasing it after this. Because 2 or 3 years after the original study, we ended up getting the whole genomes analyzed. We have a pie chart of everything there on those genomes. And part of this study is going to be to also run a couple more samples to conclude the other. We have pie charts and everything that shows exactly what's in them now. We had bioinformatics done. They were compared to everything in GenBank and other. There's another depository too that we also looked at. So we know what's in them now. We know that naturally it should not occur, but it's there, and they don't follow the rules of general hybridization because normally, like I'll use a horse as an example, a horse and a donkey make a mule. A mule is sterile.
Yep.
Same with ligers, which are lions and tigers. They'll mate and may produce offspring, but they're sterile. But we all know that these are not sterile, that it works. And we took the paper even further. We actually took the DNA and did electron microscopy on it. And it was weird because we had nice, long, healthy strands of DNA, but there were what they call single-stranded gaps. Anybody that knows anything about DNA knows it's a double helix. It has 2 strands. And it looks like a ladder that you've just twisted. So they call it a double helix. Well, we had like a single strand for long places in their DNA. And what that came from was the hybridization, because it didn't have an aligning sequence to make the double helix. So it was just single there for, several thousand bases.
So it looks like somebody cut the DNA strand and spliced in pieces where they wanted them.
That's why I said it's basically genetic engineering, but it's beyond what we've had technology for until very recently. And these things are thousands of years old. So
right.
Whoever put it together has more technology than we do.
Right. So either you're talking, other than humans or vanished human civilization or aliens, but somebody a long time ago was playing with their mad scientist genetic tinker toys, and they created this, which would make you wonder how many other of these cryptids that have been reported for hundreds of years are the result of the same sort of tinkering and chimeras and all that sort of thing.
I want you to consider this. Now, we have dogman DNA. I've got one full genome already, and I've got 2 samples that are good samples, and they both had human mitochondrial DNA. And those results will be coming out when we finish the second genome. But what's interesting is if think about Greek, Roman, Egyptian, and Sumerian mythology. It's really not mythology because these creatures still exist today. And if you think about it, there's goat men. Pan is a goat man.
Yep.
You have to look at the statues over there. The ancient statues that you can see in Rome or Pompeii. you know,
I can show you a picture of a satyr. A woman that was a fan of my show, didn't want to be identified, sent me pictures. This weird thing was following me through the forest for about 45 minutes. And as I was just about to outrun it and get out of the forest, I spotted it sitting on a rock, took a picture. It's a satyr.
Yeah. And there's one that lives about 10 miles from here that, I mean, some of the high level people in the community have seen it.
Mm-hmm.
And it's definitely a goat man or a satyr or whatever you want to call them.
Yep. They also call them fauns, F-A-U-N. That was another name.
Yeah, I mean, there's a number of names for them. Pan is the name of the god.
Yep.
But Anubis is the name of a dog man. And then there's also the, you know, I mean, really, you can pretty well take care of every cryptid if you go back to mythology. So at this, I truly believe, since these things date back to before the flood, that these things would appear to people. And since they can, and there's a lot of reports of what they call woo, of supernatural things being able to be done by these things. Now, people saw it back then. They would turn them into gods. I even have a saying for it. My saying is, show mankind a little supernatural, and they will run after it and worship it.
Mm-hmm.
Because they can't explain it. I mean, I've seen things I can't explain. I'm not going to go into it, but I've seen a lot that I can't explain. So with that said, I believe that it all ties in through the ages. I mean, because we actually in the paper have a picture of a coin that has a Bigfoot on it from 500 BC. So, you know, there had to be interaction somehow with something that either had technology or whatever to make these things like they are.
Let me give you three quick literary historical examples. The story of Beowulf is the oldest story in the English language still extant in its original written form. What does it have in it? A cave troll named Grendel that doesn't like noisy neighbors and comes out and beats the snot out of them. Sounds a lot like a Sasquatch. Then if you go back further, you've got the Epic of Gilgamesh. And Gilgamesh is this giant who's 15 feet tall, and he goes around and stomps monsters' butts. And the locals tell him that there's a scary thing that lives near their area. He hasn't actually hurt anybody, but he scares people because he's big. Can you come and scare this monster away too? Well, yeah, there's a couple different versions of this, but the one I heard, basically, this wild man basically defeats him, but then becomes his friend and his sidekick and goes on all these other adventures with him, killing monsters and stuff. And when his sidekick, Enkidu, the wild man, finally gets killed, then he goes in search of his final quest to try and find immortality so he won't die too like Enkidu did, because he's, you know, his fighting buddy died. Oh, well. You can go back even further in time than that. If you look at the Sanskrit epics, the Ramayana talks about when Lord Rama's girlfriend was stolen by Ravana and was taken to the island of Sri Lanka, which was named after the demon god Lanka. That was his island. And he had a whole army of giants that he was using to guard that island. And Rama didn't have any way to really invade. He didn't have a navy. He didn't have enough vimanas for an air force. So his buddies built this huge, long stone bridge spanning from the Indian continent to Sri Lanka, which is supposed to be about 10 miles long and half mile wide, which they then used to invade. And his buddies that built this bridge for him also, you know, really wanted to help invade, kill all the giants. They were the Vanara. The description of the Vanara is they're Sasquatch, essentially. So this is not only goes back at least 6,000 years, because guess what? They did a satellite survey of that section of the ocean, and the remains of that bridge is still there. And they sent archaeological team down to look at it. And after a couple years of digging and looking at it, they concluded two things. First of all, not natural. And secondly, at least 5,000-6,000 years old. Yeah. So that goes back to even before that. And this was, you know, one of the demigods that were running around pulling the strings at that time, had a run-in with the giants. And who did he get to team up with them? The Sasquatch. And they were more than happy to build a bridge so they could go invade and kill all the giants. So there you go. It doesn't matter where you go back in history, you're going to find that there's been stuff written about them. You know, even in Europe, In Germany, they've got Der Wildermann. In Britain and the Isles up there, they've got the Woodwoes. You know, all over Europe, everywhere you go, there's names for them.
Oh yeah, they're all over the world. There's no doubt about that. And there's other things too besides, like Dogman. Let's take it back even further. You got the Book of Enoch, which talks about. It's an apocryphal book, but it talks about about the Nephilim and how they were formed from, fallen angels made these things. And they contained animals and people and everything else. So, you've got all of this historical background on it, and it all, kind of corroborates everything, which is interesting. And talking about Dogman, there's also a YouTube video written by a monk hundreds and hundreds of years ago, where he writes about a city of dogmen. And it's it's almost scientifically written. And
Marco Polo talked about running into them too. And then there was the Catholic St. Christopher that was supposed to be a dogman that converted. And there's lots of old tapestries and illustrations from the olden days that show where these kings would hire these dogmen soldiers because they were incredibly nasty mercenaries. And they would have, if they could afford them, they would have them in their armies because they're pretty much guaranteed to stomp the enemy's butt 100%, although they charged a lot. So all of this kind of stuff is all in these old histories. There's even stories in North America from a couple of the native tribes here that at one point they had tribes living near them that were these dog-headed people.
Yes, they exist. And I've seen one. My farm help had a close encounter, and he was a military man, and it scared the wits out of him. And that was unexpected because he was pretty fearless. He had been in war and everything, and he still, he was shaking when he came up and told me what he saw. So, they've been around for ages, and they continue to be around. All of these things, not just the Bigfoot. So that's why I'm very interested in doing the study we're doing now, which we're going to do some more Bigfoot work, and we're going to finish up the Dogman samples. If anybody out there has any good hair samples from any of these types of cryptids, any of them, or even a merman or a mermaid, any type of strange creature that they're pretty sure it's got a good chain of custody, kind of to the point that they are sure what they have. We would love to have a sample.
If you find a Thunderbird feather, bring that sucker in.
Yeah, exactly.
I want to see that. There's enough people that I've seen one of those things. Obviously, they exist. Somebody grab a feather from one of them. They're black.
Yeah, because feathers do contain DNA. You can't extract a feather. So, teeth or hair are the best two options on any of these things. But, we would like to tie this all together because my theory is they're all going to be related. They're going to have human mothers. I think they're all going to be a hybrid of some sorts.
So, how many Sasquatch DNA samples did you actually test? It was over 100, wasn't it?
I think it was 106 or something like that. It was over 100, and then we have more that came in after the study. I'm going to do a couple of those, just confirmation with the hair.
So every single one of them across the board, then, if I'm understanding this correctly, came across as hybrid with human female. None of them were like pure blood, although you don't know exactly what that would be anyway.
Right. No, all the samples we tested, the mitochondrial DNA was human. And they were mostly Middle Eastern or European. We only had a few outliers. Everybody thought there'd be a lot of Native American, and it wasn't. We only had one or two, I think, that were Native American. And y'all can go look at the paper on SASQUATCH and do the count. It's been a few years, so I haven't, but I'm close on it. I can tell you that much. We had a couple of African samples, but all of those were outliers. And we even kind of had a theory. When you got a hybrid, you don't know exactly when it happened. So one thought is that, say, one of the African ones could have been like a slave that escaped and ended up mating with a Sasquatch, because there's a lot of stories from the Pacific Northwest where they would, and I know a native chief up there that I mean, he knows. He has relatives that are part Sasquatch. They used to take the maiden. And
yeah, I know. I heard that there's like 2 or 3 tribes up there that didn't think that it was like a bad thing for a Native woman to have a Bigfoot husband.
They won't let anybody have any DNA. It's a shame because that would be huge because we could, follow it down through the generations. And he even knows where the daddy Sasquatch is buried, but they won't reveal it. So it's a moot point.
How far back do you think? I think what you're getting at is there was a progenitor, a pureblood at some point that hybridized. How far back was that?
13, in general, 13 to 16,000 years, generally. The African outliers were 25,000 years. But once again, I'm not so sure that that's when they actually came into existence, because that could have been at any point in history where, perhaps a escaped slave or something had been hiding in the woods and ended up incorporated into a clan. So that's very definitely could happen, because one thing about hybridization, you can only guess whenever as to when the hybridization occurred, and you can only do that by the amount of say human DNA in it.
Right.
I mean, but after just so many generations, it's going to change and lower. So anyway, it's an interesting theory that I think they're all interrelated, and that's what I want to prove. But also, it will just give us more information about what the makeup on all of these things are. And
well, keeping in mind that a lot of their presuppositions on who was where and when they died out are pretty much guesswork, because they've recently found out Homo nodali down in southern Africa, they had presumed got extinct way before modern humans were in the area. And they found some remains in a cave that were like not that old, so they were wrong about that too.
Oh yeah, no, I'm not dead set on the timing, but you can do it fairly well, because what it's based on is, say, a mutation every 1,000 years or something, and so you can kind of trace back the different mutations, and that's how they kind of age the DNA.
What really strikes me from what you just told me is that. Oh, go ahead.
I said it's a rough guide as to how old they think the sample is. You can argue it both ways, but let's just say one thing it will prove is they're not ancient at all. Ancient would be, well, they have to have existed from the time that we have modern humans for sure, for the simple reason that their mothers are modern human.
Yeah, and how far back does that go, though? That's another thing that keeps getting extended all the time, is the timeline on how old are modern humans seems to go back way further than what they're willing to admit.
Right, but who knows if that's right or not either. It's still theory.
Yeah.
And I don't think we'll ever really get to the bottom of that, but you can use kind of a rule of thumb and get an idea. But let's just say that they couldn't have existed before us. And there's a lot of people out there now saying, oh, they're ancient peoples, they were before us. Maybe their progenitor, their male progenitor, could have been before us. But the actual Sasquatch, since they're human hybrids, they definitely are younger than the human race.
Which brings to mind 2 questions. First of all, you mentioned the African outlier, so let's not bring that up. But in this general discussion, you're talking about hybridization happening like 16,000 years ago. That's the height of the Ice Age. There are lots of animals not making it through the Ice Age, and there could have been other things that we're not aware of because the geological record isn't complete enough. It was too close to the surface. We know that there were some major eruptions in North America about 5,000-6,000 years ago. We also know there was a meteor that slammed into northern Greenland about 12,000 years ago, which could have been what ended the Ice Age in the first place. And then they've been trying to say that the North Americans came across the Beringia land bridge during the last Ice Age, which is just recently proven is complete hogwash. And they found 23,000-year-old tracks in Arizona in the rock. So a lot of the stuff they talk about doesn't line up. But here's what I'm wondering, if at some point, and we know there's been bottlenecks, like humans almost completely died off when the Toba eruption happened. They figured no more than 1,000 mating humans survived that, pairs. to continue us on. We almost died off completely. So being that there's these massive catastrophes at fairly regular intervals, is it just possible that during that last stressor period where a lot of the species were dying off, that's when the progenitor, whatever it was, actually started hybridizing with humans, and that's where the modern Sasquatch came from?
Well, that's probably as good as anything. Or you know, some people are going to believe that aliens have created them all. Others are going to believe the biblical account that you know, they were formed. They were originally the men of old, the men of renown in Genesis 6, and then, you know, as it progressed, other things were formed. So there's a lot of different theories out there, but every one of them, the one thing that comes from all of it is we were before them because they have human mothers.
Mm-hmm.
That's the point I want to drive home, because there seems to be a lot of people thinking that they're ancient peoples that were before us, and that's just not true. It can't be, because they have human mothers.
Right. The current ones, what about the progenitors, though? I heard through somebody that's connected inside the deep state and was collecting DNA samples way before you did a study, by the way, for NATO, that they had already decided that most of the Sasquatch were hybrids, but they also said there was a pure blood, and apparently it's still around wherever the hell it is.
Well, I wouldn't doubt it, because, you know, I mean, there's a lot of things. I mean, if you want to go into some weird science, you know, rumor has it that they found the remains of, you know, the chained fallen angels, allegedly. And if you believe the biblical account, that could account that yes, there's still a purebred around that would have been a male progenitor.
Mm-hmm.
But, you know, I'm not. People are going to believe what they're going to believe. I'm not trying to convince anybody, but there's so many different ways to look at it. And I never say never, because I used to say never. I used to say they never would exist. And now I've learned so much that I say the older I get, the less I know, because I keep learning new things. And
yeah, for a long time I didn't want to believe in any supernatural hokey pokey of any kind, but I keep seeing more and more examples of it, and it gets harder and harder to deny it. And at some point you're just lying to yourself, because you're seeing clear-cut examples of strange things going on that we cannot clearly identify, and we don't have any kind of borders on what is this phenomenon. And that's how it gets lumped into the category of paranormal, supernatural, or woo, because we don't have the scientific mnemonic tools to describe the phenomenon or understand the mechanics of what's making it happen well enough to describe the phenomenon. So it's magic hokey pokey, because our scientists can't describe it.
Well, scientists generally don't believe in anything of any sort of supernatural.
Right.
Most of them are atheists, actually. I know, because I was amongst them.
Right, but what I'm saying is some of the stuff they claim is supernatural, you go do the scientific tests on it. You know, you have a theory, and you come up with a way to test it, and you do the same test over and over again, and keep getting the same result. It's not supernatural anymore.
Well, you know, with all the reports on the Sasquatch, the DNA is there. They're a physical creature.
Yep.
But they also are like nothing that else is on this earth. And I prove that with the fact that the hair does not yield DNA. If you take a mammal and you cut off a chunk of the hair shaft, you're going to get DNA from it. I mean, if it's a brand new born baby and the hair is so fine, there may not be enough DNA to show up. But let's just say generally, with any species on the planet that's a mammal that has hair, you're going to get mitochondrial DNA out of that hair shaft. You cannot do it with Sasquatch. We spent 3 months troubleshooting that. 3 months. And we changed reagents, we changed protocols. You know, my second author test, they had a state-of-the-art robotic system, and they robotically extracted. And it came down finally, we put it on the spectrometer, because we didn't know if there were inhibitors in the hair or whether it was just no DNA. And there was no DNA in that hair. And it had a medulla, it had everything it should have had to have the DNA. But yet it has no DNA when you try to extract it. And, you know, it's the only animal slash primate slash hominin that I've ever heard of that does not give mitochondrial DNA. I can, you know, and I'll make the caveat about some very fine baby hair, but basically, this is coarse hair. It should have given us gobs of DNA, and it didn't. And this is bizarre. And it's in the paper. And they did this psych study. And they actually take a swipe at me saying contamination, which is, I've got the graphic proof there is no contamination. But that's beside the point. They had 7 samples that didn't run.
They also had some samples that they had Robert Kreider go out in the field and collect for them and send to them that they didn't bother to test. Well, and he also said the Yeti hair that the Yeti hunter in the Himalayas collected from inside of a hollow tree was a formerly unknown species of polar bear presumed to be extinct for 20,000 years, which, by the way, a survey of all the bear DNA from around the Himalayas just came out and said that's complete and utter bullshit. Sykes is a fraud.
Well, bless his heart, he's passed on now. Don't want to talk bad about the dead, but
I don't care. I talked bad about him when he was alive. I'm not changing my opinion now that he's dead.
Well, I'm going to criticize the paper, though. Within 6 months, Cambridge had another paper published about the bear thing. So he was wrong on the bear. But what I found interesting was the 7 samples that and he couldn't figure out why, even though he had read my paper, because he had contacted me. 7 samples did not yield mitochondrial DNA, and all they used was hair shafts. Those 7 samples came from people from my study. So we had tested those samples. We knew they were SASQUATCH samples, but they didn't use the hair roots that you have to test for SASQUATCH. They used the hair shafts. So those 7 samples were from my study, and they didn't run, and they wouldn't have run. It just supported what I already said. So it's kind of interesting. And then they try to get me on contamination, which I find hysterical. So, you know, the paper, and I'll tell you something else that's really interesting about that paper. They credit the Fish and Wildlife lab here in Ashland, Oregon, with screening the hair samples. Well, let me tell you a little something. I was an invited speaker to speak on animal forensics when they put on a forensics meeting up there. And I met those people, including the director. And whenever I found out that he was doing this and they were supposed to be screening, I called him. And I said, hey, you know, I already did a big paper on this. Why are you just going to do the mito on this? And he kind of argued around. He says, well, I'm just going to do it. I want to do it. Okay, fine. I said, well, I know you got bodies. Not here, we don't. I said, yeah, but y'all got bodies. I know you got bodies. He says, you know, I can't talk about that. At least he didn't lie. Mm-hmm. But when the Sykes paper came out and they credited it, let me tell you something. They had wolf and everything else in there. And the wolf hair is so easy to tell because it's got kind of a spoon-like thing on the end to shed water. And whenever Sykes was testing all these samples, they had all these different species. Those are the best hair analysts for animals in the world at that lab. They would not have missed all those samples. So either it was a put-up job or else they never screened them, even though they were credited with screening, and the director told me they were going to. So, you know, the whole thing is just suspect in my mind.
Again, Robert Kreider was picked to go out in the field, find fresh Bigfoot hair samples or DNA samples by the local university, not knowing who they were connected to. Went out there, did it, documented the whole chain of custody. I've shown this on my show. We got the video of everything all the way down the line, including the positive air pressure system that he's using to take them from where he had them to put them into the other container and label it and the whole thing. And he sends it off to Dr. Meldrum, who then claims that he loses them. You ever heard of a chain of custody, supposed doctor Meldrum. Well, as it turns out, he actually did send them over to Sykes, which he was told specifically not to. And Sykes not only didn't test them, he sent them to Luzon, Switzerland, where they're still sitting.
Well, that's the whole thing. The government's behind the cover-up. And nothing's going to get done unless you do it privately and, do it kind of how we've done it. We did not tell these labs what they were testing. We just paid them and said, we need these samples tested, and thus and such is what we want done.
That gets a more accurate, unbiased result anyway. They have no idea what they're testing. Let's see what you think it is. Yeah,
and thus the letter saying, have you discovered a new species? And so, it's really been an interesting ride with all this, and the way all of the many, many journals would not take us at all. Wouldn't even consider it after Nature took it. And only one in 10 papers in Nature actually even makes it to peer review. And we made it through the first peer review and still, and then things changed. Mm-hmm. Somebody got a hold of them is what happened. And so, and then I've had people that have worked with me that have been threatened by the government. We had I'm sure it was a government operative sitting beside our lab at one point. And when we went out to try to talk to him, boy, he took off. So, it was a small town, so everybody knows everybody. And this guy stuck out with a sore thumb. And where he parked, nobody would ever park. So it was really strange. So I know that there's cover up. And then there's people that are haters that, they're jealous. They want to be the first one to discover it. Well, I got news. I'm not even the first one. I'm just the first one to go public with it.
The guy that developed taxonomy, Linnaeus, actually classified them. They were discovered a long time ago.
Oh, yeah, but I'm talking some real science.
That was the only science there was. He invented taxonomy, and he said, these things are real. Here they are. Here's what we call them, Homo ferus.
Yeah. And this is something that I'm going to leave a teaser because this will come out with the new information. I had a whistleblower that was dying contact me.
Okay, hold on. First of all, we got to take a break because if I keep trying to run the program much longer, it's not going to record. It's going to crash. So we're going to stop recording.
Okay, this is the video everybody's been waiting for. This video is to show how we knew we did not have contamination whenever we did the Sasquatch Genome Project, when we did the paper. We're going to talk a little bit about the peer reviews and how they kept saying that the samples had to have been contaminated, and so rejected the paper on those grounds. And then we're going to show you how we could determine with multiple types of testing that there was no contamination in the samples. And I'm going to show you actual data, some actual data and some data that I had to actually put together to show contamination since our samples weren't contaminated. So we'll go through each of these slides and I'll explain a little about it. We're going to talk first of all a little bit about the Nature reviews and about how the methods that we use to remove contamination prior to extraction of the DNA. I don't have a video of me washing the samples, but we have a little vortexer that is extremely vigorous and rapidly moving. And what you do is you put an alcohol and water combination into onto the actual samples, hair in this case, and you vortex them with this professional vortexer, which will shake loose any loose cells, because that's where where you touch something, you'll leave a cell which contains DNA, and this shakes loose any cells. And we did this multiple times per sample to make sure that there was absolutely no foreign DNA on the hair, that the only DNA that was there was in the actual hair. So we did this to every sample we could. Now, of course, there were some samples in the study that we couldn't wash, those being saliva samples or other liquid samples. But there's not a problem with that, because you can always tell if they're contaminated or if it's a mixed profile whenever you move forward into the actual DNA testing. The contamination would have been visually obvious when doing the DNA testing using, 3 different methods, and we used all of these when we did our testing in the manuscript. Sequencing using universal primers as well as standard sequencing, and we'll get into how this relates in a minute with some pictures. We did Powerplex 16 screening of SASQUATCH samples, which is your forensic profiling with STRs, and we'll get into that in a minute also. We also used negative and positive controls to assure ourselves that no laboratory personnel had contaminated the samples. And if they had, we would have been able to determine who had contaminated it in the lab, because every lab person in a forensic lab has their profiles on file, so that if there is a band in the negative control, you can determine if it came from a person in the lab. Not only that, negative controls, if you haven't if you've got a chemical or something that's contaminated, regardless of who or where it got contaminated, it'll amplify and show up as a band in your negative control. And you do more than one. You do what's called an extraction blank, which means that you have your solution that you put in when you extract your samples, and you run that in a lane and run it through to see if there's any DNA showing up in your extraction buffers, first of all. And then you have also a negative amplification control, which means after you've done your PCR, you will run a PCR along with your other samples that doesn't have any DNA added to it. And if DNA shows up in that negative amplification blank, then you know that something in the amplification is contaminated. And you also use a positive control to show that your PCR is actually working, and your extractions are working. So with that in mind, we'll go to the next slide and talk about Nature peer reviews. There were 2 rounds at Nature. The first round was better than the second one after we did so much more work. Reviewer 1, on the first review, he actually passed the paper with revision. He said it was Nature worthy, but it needed to have some revision. And he gave a lot of small corrections, verbiage or things that we said he didn't like, and we corrected all of those and responded to his review. In fact, we responded to every review we received from each of the reviewers, And if you look at the bottom of this slide, you can see where to go actually read all of these peer reviews, including the passing reviews we got from the James Journal before acquiring it along with those peer reviews. Reviewer, back to Nature Reviews. Reviewer number 2 on his first review, he didn't read the manuscript. He asked us to use universal species identification test on the mitochondrial DNA, and that was already in the paper and it had already been done. He didn't ask to see any of the electropherograms, the graphic representation of that the testing was done. He also asked for whole mitochondrial genomes, and we already had a number of those in the paper. So he didn't read it or he would have seen them. He also wanted more nuclear DNA, so this triggered us to sequence the 3 whole genomes prior to resubmission. Reviewer number 3, his first review was unprofessional. I mean, it was laughable. He's obviously not English or American or any English-speaking country, because his English was very poor, and I quoted it on this slide and left it exactly as it was written. And basically he's saying that that our testing showed that a Caucasian woman had to run around in the forest having sex with a hominid and left the baby to it. And so he was making fun of us. He didn't even give a serious review, and I complained about this to the editors at Nature. So he was professional in his second review, but still, as you can see, we'll address that on the next page. Reviewer number 4 was more professional. He made a list of grievances, and one of the things that got me about him was he said, "Oh, the hair analysis has to be wrong." He says, "But I'm not a hair analyst and not an expert in it." Then you shouldn't comment on it saying that the analysis is wrong when you're not a hair expert. Only a hair expert can say that. He did make a valid point about wanting more data, which we gladly provided by sequencing the 3 whole genomes prior to resubmission. However, none of these people asked to see any of our raw data, which normally they don't. I mean, when you submit a paper for publication, they come in and they read it, and I don't know of a case where they've asked to see the raw data. But if they were going to call contamination, they should have asked to see the raw data, and they didn't. On the second submission to Nature, suddenly the reviewer number 1 suddenly says he's not qualified to review the paper after he's already reviewed it. And he says he'll go along with whatever anybody else says, the other reviewers, that, he, even though Nature sent it to him and thought he obviously was qualified to review it, after one review he decides the second review that he's not qualified, so he'll just go along with whatever the other reviewers say. So reviewer 2, now, he was the one that didn't read the manuscript the first time and asked for all these things that were already in it. The second review, he still didn't read the manuscript. He asked for materials and methods which were stated in the manuscript to be found in a supplemental document provided. It's funny because the paper was so long, the editor actually asked us to put the supplemental material, the materials and methods in a supplemental document because it was so long. And so we did that, and then he says we don't have it, even though in the actual manuscript there, and you can go read it for yourself, we say look to supplement whatever to find the materials and methods. He also kept saying that the samples were contaminated and or degraded, which means that they were basically had decomposed, which, I can address that in another video. We actually have a figure in the paper showing gels that would, if it was degraded, there would be a smearing of the DNA instead of a nice, neat band. And so we provided those already in the paper that the DNA was not degraded. And he kept calling the samples ancient DNA. And we couldn't understand this because the samples were basically fresh, especially the Erickson samples, because they were taken directly from the field and put into a plastic bag and into the freezer. So they were extremely fresh. And the hair, the hair follicles, if they had been degraded, they wouldn't have tested, and they would have also showed up as being very smeared on our yield gels. So that picture's already in the paper. You can go look and see. Reviewer 3, remember, he was the one that made fun of us and was so unprofessional. This time he just said that it must be a mixture of animal and human DNA, and it, had to be contaminated, and he just, didn't believe it. And that's exactly, what he said. Once again, at the bottom of the page in red, you can see the actual place where all of these reviews are, and you can go read them yourself. Reviewer 4, he refused to review it at all the second time. So we're down to 2 reviewers that make the decision. One didn't read it, and the other one made fun of it and said he didn't believe it. It was just had to be contamination. Did they ask for any proof that we could show that it wasn't contaminated? No, they didn't. They just, blew it off out of hand. So if this isn't professional bias, I don't know. But at the end of this video, I'll show you the ultimate bias. It's laughable. Okay, a little bit of science, and I know that some of you are going to have a little trouble understanding this, but I'll try to make it as easy as possible. There's 2 different types of DNA testing that we used in order to determine whether or not there was contamination in the samples. Since there was no contamination, I'm just using some stock slides I had to show this. On the left, you have what's called STRs, and these are lengths of DNA that are inherited. They're not in the genes, they're not per se, and they're not in a coding region or anything of the DNA. They're just little repeat sequences that happen whenever when 2 parents have a child. The child is going to have a combination of those length sequences. It's inherited. It's used for paternity testing, for forensic profiling. It's got several different uses. And I have a very simple. This is actually a horse set of electropherograms, which is what you call these graphic representations of the DNA. And there's several different markers here. There's actually, a space between them so you can tell where they are. There's either 1 or 2 different peaks to each of the loci or place where the DNA is. Loci is just like the location on the chromosome where you find it. So I gave an example on this of if you have an individual, and they have names they give to these, but we're talking raw data here, so we're just doing it in numbers of base pairs. And a base pair is the 2 nucleic acids that make up each point of the sequence and the length of the sequence. So you have 168 base pairs over 184 base pairs on one of these loci, and you can actually look, and it will be, let's see, I don't believe we have actually that one. This was just an example I made up. 184 repeated bases from one parent and 168 repeated bases from another parent at the same spot in the DNA. Therefore, it's called a short tandem repeat. These are actually dinucleotides, meaning that there's 2 bases for each repeat. So, like for instance, your DNA sequence on a length of repeated DNA is like CA, 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 as the example below. So these, like I say, these lengths are inherited. So where you have, one single peak, it's like both parents gave the single peak or the one same size length of DNA to the offspring. Like in the top, the real tall blue peak to the far left is basically a homozygous peak. It means that it looks like it's about an 89 over 89, which means that this horse inherited an 89 length at that locus or that position on the DNA from both its parents. If you come over to the there's a second single peak, and then there's a double peak. The double peak there the parent would give one peak of one length of DNA to it, and the other parent would give the second one. So it's called heterozygous. Probably an easier example to see is on the green, there's a single peak, and then there's two peaks that you see there. Those two peaks are once again heterozygote, got different length of DNA fragments from each of the parents. So it's called a length polymorphism, or a change in length of the DNA repeat. And repeat is once again just the same nucleic acids repeated over and over, like CA, 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 CA. The second type of DNA we're looking at is what we call SNPs, which are single nucleotide polymorphisms. They have to do with variations in DNA sequence from one individual to the next. They're little mutations, single base mutations. And once again, you got an example here. You'll see that in the where the era is, the upper electropherogram has, or chromatogram as some people call them, has two red peaks, and you look down to the bottom at the same place, and you got a red peak and a blue peak. So the second peak there, one of them has a thymine, and the other one has cytosine, which means that they are different at that particular locus, and therefore it is a sequence polymorphism. There's a single nucleotide that's different from the top individual to the bottom individual. So that's an example of when you're sequencing, you can see the difference between individuals. We'll talk about sequencing first. The peer reviewer asked, wanted us to use universal mammalian primers. And if you wonder what they are, they're short sequences or pieces of synthetic DNA that are synthesized. They're artificially made, and they're made to attach via enzymes called polymerases in the polymerase chain reaction. They target DNA in order to select particular sequence that you want to replicate when you use the polymerase chain reaction in your amplification of DNA, because you have to amplify DNA in order to attain enough of the DNA material that you can visually see it when you put it in the machine, in the sequencer. So I have a little graphic representation there. I'm sure it won't mean a lot to some of you, but it shows how the primers will attach to a specific spot on your DNA, and it will cause that particular place in your genome to be amplified to make enough copies so that the sequencer will show it. Universal primers simply mean that they work in many species, in this case mammals, and there's several published papers on different. There's cytochrome B, and there's ones that they're in the I guess the common name that you find if you go to query them is DHL and THR is the name of the two primers. And they're in hypervariable region 1 of the mitochondrial DNA. Now, both of these types of universal primers, we use both types. They will, when you take the sequence that they generate at the end, and you blast it against GenBank, which is the international depository for DNA sequences, there's just millions and millions of them in there. It'll tell you what species you have using the mitochondrial DNA. And this is how species determination is done when you have like a forensic case where there's animals involved. We had a kind of an interesting case where some dogs had killed some horses, and they swabbed the horses for the dog's saliva, and then they impounded the dogs and collected their feces. And we ended up testing those samples to determine which dogs were guilty, leaving their saliva on the victims as well as passing equine DNA in their stool, and which ones were negative for this. There were like 6 different defendants in this case whose dogs were blamed for this, and I think, if I remember correctly, 4 of them were guilty. Anyway, the universal primers are synthesized with the idea that, you know, certain parts of your DNA are shared among a lot of species. And so if you put your primer in the region where it's shared between many species, then the DNA that's in between those areas will be amplified, and you can, you know, tell what species you have. Anyway, all of these tests are just standard tests. They're in the literature. We didn't do anything special in order to do these tests. They're easy to show contamination with using these tests, because generally when you manually sequence, you're gonna, at least when you're doing it for forensics, you do what's called a yield gel, and you put the amplified DNA from these universal mammalian primers onto your yield gel, and you've seen pictures, I'm sure, on TV where they put they have all these little holes, and they're putting the colored liquid into the little holes, and then they migrate forward and they form these bands that are so popular on these forensic shows. And these bands of DNA show, A, that your amplification worked well, and B, when you're using universal mammalian primers, a lot of the species have different size fragments, so you can see if there's more than one species in your sample. Here's an example. Now, I had to make this example because we didn't have any contaminated samples in the study. We had, in this slide particularly, I used dog DNA and I mixed it with human DNA. This is using universal mammalian primers. We had a human positive control, and then we showed a negative control there. There were actually 2 of them, but I sliced it up so I'd have room to put the mixed samples. There were 3 human samples, and then there were 7 dog samples that we ran on this yield gel. And then I mixed human and dog and put a human between them, and then human and dog again. As you can see, the dog sample band is a different size than the human, so you've got 2 bands showing up, and this would indicate a contaminated sample. You can actually see more than 1 band present, and that shouldn't be. If you have a clean sample, you're only going to get the single bands. They may be at a different size and show and not be at the same level when you're using these mitochondrial primers, but you will see a single band unless you have a mixed mammalian sample. Well, obviously SASQUATCH has hair, so that would make it a mammal. So these are a good type of test to use. Because most likely, if there had been, you know, like a dog or a bear or something in the sample, it would have shown up different than the human that we got the results from. Okay, now here's an actual yield gel from the study. This was using the hypervariable region 1 primers, THR and DHL, which are universal mammalian primers, and we use these commonly for species identification in forensic cases. You can see the little light bands at the bottom. Those are just leftover primer bands. You see those in these gels. They're just artifact and not to be worried about. And they'll be a little brighter whenever they're not used up in the DNA, and you can see that with your negative controls. The first lane shows an extraction blank, a negative control. This was the extraction liquid with nothing done to it, and there's no band. And then you see the 3 Erickson samples, 37, 29, 31. 31 had the whole genome done, and you can see that nice clean bands. There's no other bands around them. There's no contamination there. Strictly human. And this is in mitochondrial DNA now. and then you have an amplification blank, which means that this was our amplification mixture. The liquids we use with the enzymes and everything to perform the polymerase chain reaction where you amplify the DNA. And if there had been any DNA contaminating any of those reagents, or if we had handled the samples inappropriately, there would have been a band present there. And then we have our human control, which obviously is nice and bright and on the same level. This shows that there was only one species, and it was human, in the mitochondrial DNA of these Erickson samples. Of course, this was preliminary. This is before we even sequenced. Now, here's the graphic representation after sequencing that the machine puts out. There's 4 nucleic acids present, and they're each designated by a different color, and that gives you your DNA sequence. You can see the abbreviations for them at the top of each of those pictures. Cytosine, adenine, thymine, guanine, the 4 nucleic acids are abbreviated by the first initial, and the order that they're in is what makes you an individual. And so these are 2 different individuals, and in this short little piece of sequence, there's 6 different polymorphisms, or SNPs, or single nucleotide polymorphisms. These are mutations where the top individual is different, or a different individual than the bottom individual. And the errors point to where you have these individual differences in the sequence. Now, when you have contamination, it would be like one of these was with an overhead projector. In the old days, you could have a clear graphic and you could overlay it on top of another graphic. Well, this is what happens when you have contamination with sequence. If we took these particular 2 and overlaid them, you would have 6 places in the same species in a short little stretch of DNA where you would have.
Well, one East Texas woman has dedicated the past 5 years to studying DNA of the species. Members of the SASQUATCH Genome Project recently released new, higher quality, never before seen footage of Bigfoot. Dr. Melba Ketchum tells East Texas News 9's Leah Hughes that Bigfoot is real, and her research proves it. It's new at 10.
It took the SASQUATCH Genome Project 5 years and nearly $500,000 to do it, but this group of scientists believe they have proven the existence of Bigfoot.
Neither evidence will stand alone under the scrutiny we're getting. Well, mine would, but it's not interesting to viewers because it's DNA.
The group recently released this new video they captured of the elusive creature. Lead investigator Dr. Melba Ketchum says she studied a DNA sample of the specimen you see right here in the video, shot by team member Adrian Erickson.
We tested, among other things, the red hair gene. And when we sequenced the red hair gene in this individual, she's red, and she had human sequence at that particular gene, and it has red hair. The physical attributes of the specimen in his video is exactly matches what we tested, and nobody could have predicted that. In other words, her DNA matches her video, which is so wonderful.
Ketchum says she has no doubt that what she is researching is some kind of human hybrid.
They're more whatever they are with a little bit of human.
But you won't catch Ketchum calling them Bigfoot.
They're humans. You're type of human, you don't want to kill them. That's wrong. We want them protected. They don't need habitat. They live fine like they do.
And when asked why there weren't any remains or bodies to study,
they are able to hide from us. That's why there's not bodies. They live in an environment. They have the cunning of animals, and they have the intelligence of human and more.
Ketchum says she was once a skeptic, but all that changed because she has seen Bigfoot with her own 2 eyes.
So they took me up to North Texas, and I was able to actually, I saw 5 that day, which just blew my mind.
You were 100% certain you saw what you saw.
Oh yeah, there's no doubt in my mind.
Ketchum says she hopes the new video, along with her research, will result in some recognition from the scientific community. Leah Hughes, East Texas News.
Well, Dr. Ketchum says there have been numerous Bigfoot sightings right here.
We are back again with the amazing Dr. Ketchum and continuing on with our conversation about Sasquatch DNA and the ongoing cryptid DNA study. And we left off on the subject of government suppression. So welcome back again to the show, Dr. Melba Ketchum.
Thank you. Yeah, the government doesn't want this out. That's been quite obvious with the suppression of public paper, the threatening of people that have worked with me, and them having an agent out by our lab, among other things, when we were doing the work. So, with all that said, yes, it's a major problem. I think they also have trolls out there that, well, I know they do, that not only say untrue things about me, but they just generally cause a lot of problems. And they put stuff out that's just not true to make me look bad and scream contamination and all this, that, and the other. And I've had death threats and everything else. So they're probably behind some of that, at least. Of course, there are the legitimate haters. They want to be, quote, the first one out there. I mean, I wasn't even the first one out there. I'm just the first one to go public with it. So I can't say that I'm the pioneer of all this. I probably have the most up-to-date data because we used all the, most modern techniques at the time. And those haven't changed that much in the last 10 years, but they've gotten a lot less expensive, thank goodness. But
that's a big point right there when you want to go ahead and continue doing an ongoing study. At the time that you did this original study, it was ghastly expensive, and it's gotten considerably cheaper. It's still expensive, but it's gotten considerably cheaper to do it now. So that's going to, enable you to get a lot more bang for the buck that you can actually get anything done.
Yeah, absolutely. And, we've got all kinds of samples. We've got we're doing a few more Bigfoot samples from hair. We have 2 really good viable dogman samples that are already partially completed. We lack the whole genome on a second sample. And assuming it lines up with the first one, we're home free on that. Then we have a couple of giant samples that came from the Lovelock Cave area. And then we received them from the museum out there, and we have some elongated skulls, and we also have the famous Russian samples, Zayna and Quit. They're at the bottom of the list, though, because there's already been some work done on them. I would like to validate that work, but I want to get the unknowns done first.
Right. So what have you got from the Lovelock Cave Giants, the Seneca is what we're talking about? Have you got bones, or what kind of samples have you got from them?
We have teeth from 2 of the mandibles.
Nice.
So we have 2 samples from them. We have a great huge tooth also that we don't, it's humanoid, but we don't know what it's from, but it's giant. And that one we just found in the desert by an individual. And then we have a humerus, a arm bone.
Was the big tooth found anywhere near the Superstition Mountains by chance?
I really don't know exactly where it was found, but it was found, and so it's going to be ready to go. So we've already started. We haven't started any testing yet. We're waiting till the funding of the project is where it's going to be. We actually have met our goal, thank the Lord. We are doing really well with getting research money, but the more we get, the more we can test. We've achieved what we do to get the minimal done. And anything above that's going to let us do even more studies, which is great because we're looking for more cryptid samples. I would like to have, actually my bucket list has mermaids on it because I do believe they exist. They're a hybrid like anything else. I also would love to have, a goat man sample or a satyr or whatever you want to call them.
Yeah.
I would love, and hair is always, hair and teeth are the testing, platforms we really like because they can be washed to get any contaminants out. So, those are the types of samples that we really like. Although if it's something really interesting, we'll consider doing other types of sample, blood or saliva or whatever. But
I know you got one sample in the original study that was a toenail. That's a little bit out of the ordinary.
Yeah, but it was very sample. Actually, I've talked about revisiting that as part of this. And the more money we raise, the more of these types of things we can do. And, because it did meet all the criteria to be a Sasquatch sample. It was in the study, and it ran well. So,
that is very cool.
Yeah, it is. We've had all kinds of samples, and but I like the hair. But the hair has to have roots on it. We visited this in part 1, where Sasquatch hair shafts don't give DNA, and that is very unique because mammalian species give mitochondrial DNA from their hair shafts, and especially big viable hair shafts like these things. These are like horse mane hair. It's coarse.
Yeah.
And it should have gobs of DNA in it, and it has none when you extract it. And like I said, we spent 3 months, trying to figure out why we weren't getting DNA from these hair shafts. And finally just had to boil it down to putting them through a spectrophotometer to see if there was any DNA there at all and not just inhibitors. And it was no DNA. So that's unusual. And they have the medulla, they have everything. All the structure is there, like a normal mammal. But they don't yield any mitochondrial DNA out of those hair shafts.
Well, thanks for clearing that up because that oft-repeated thing keeps going around over and over again. Well, the reason you can't get DNA from the hair is because there's no medulla there. And what you're saying is that irrelevant, even if there's a medulla there, there's no DNA in their hair.
Exactly. In their hair shafts. Now, if it's got a root, yes. But if it's just the hair shaft itself or a shed hair, no, you can't get any DNA from it. And that's unique, period. And like I say, we spent 3 months troubleshooting before we came to that final conclusion. We were very careful about this. And so, it's real interesting. We have pictures of the hair in the study. And, you can clearly see the medulla in them. So there's no reason, there's no real reason for them not to have it other than they are just not like the rest of the animals on Earth. Or people or anything else for that matter. So
very peculiar. Did you get any samples sent to you that were specified to be from a skunk ape? Just curious.
We had some from the region that would have possibly, the one Erickson sample was from Alabama,
right?
Which could be a skunk ape.
Let me see about getting you a sample from an actual skunk ape collected so that you can test that because apparently they're not the same thing. The Sasquatch don't consider them to be the same thing. They're close to them, but they're not the same thing according to them.
This is how I look at it. There are different races, just like with people. They're going to be
Yep.
similar. They are not the same thing.
Yep.
I mean, they probably are a little more diverse than human races, but they're still basically the same thing, but there's going to be some genetic variation. So I just call them different races, for lack of a better term.
Well, that's part of the
like
problem, because even
that-
within witnesses will tell you, the ones that are actually long-term habituators and dealing with the same group and the same individuals over and over and over again, and they see the new babies being born and they watch them getting bigger and stuff, and the amount of variation within a family group seems to be way larger than what you would get out of an equal human-sized group. Like, they got a lot of variation.
I can explain that real easy. They are hybrid.
Yeah.
And when you have hybrids and recombination occurs, you're going to have this wide range of variation because, like, for instance, whenever the DNA, when the mating occurs, you know, you might say inherit it more human from one parent and less from another. Or you're going to get, you know, yet another amount of more unknown or something. It's not going to combine equally like it will in people, because we're a pure breed. Like, say, dogs. You know, there's all kinds of dogs. There's still dogs, but there's all different kinds. And, you know, but if it's a true breed, it'll generally breed true. But they're not hybridized with other things. It's like, now let's take a true hybrid, like, say, a donkey or a liger. If you have the mother to be the donkey and the stallion the sire, the creature is going to look more like a donkey, whereas a mule looks more like a horse. But even at that, you get this different recombination that some mules look more donkey-like and others look more horse-like. The same with your hennies or your female donkey bred to a stallion. You're going to see some hennies look a little more horse and some hennies look a little more donkey, because it's an unequal combination whenever the offspring is produced, that they may predominantly get more genes from a parent that, you know, it just doesn't work that way with a hybrid. A hybrid is completely different, and they can give a lot more phenotypes or physical appearances than somebody that's not a hybrid.
So you can have two parents producing offspring, and all the offspring are going to look a lot different than each other.
Yeah, they can look a lot different. And like I say, it's just a matter, you've got hybridized parents. And so they're going to have, let's just make it real simple. Say, for argument's sake, say they're human and ape. I mean, that's not what they actually are, but just so to make it simple. You've got human and ape. Well, whenever you get the gamete, which would be your sperm or your ovum, you're going to get half of that complement. And that half of the complement is not going to run true. It's not going to be like all human or all ape or whatever. It's going to be a combination of the two. So one gamete or one ovum or sperm might have more human-like characteristics, whereas the other one may have more ape-like characteristics. Mm-hmm. And then you have the other side where the other parent is going to have the same thing going on. So feasibly, you can run the gamut all the way between human and ape if they were half human and half ape. They're not, but just for argument's sake, any hybrid, you're going to have hybrids that look more one or the other and anywhere in between. So it's going to be a huge variety of individualization with that. I hope that's clear.
Yep, it's interesting and makes sense to me. So you're looking for samples of other things besides Sasquatch, and we have identified a couple of the extremely dangerous cryptids on this continent that aren't Sasquatch. And we'll see if maybe we can get you some samples from them to take a look at, too. One of them is the Gugwe, which I think is related to Dinopithecus, but we need sample to test that because it sure looks like them. And then the Genome, which is some sort of Bigfoot hybrid with something else, which we suspect is, again, back breeding with giants or whatever. So that's another one.
I want some real double-rowed teeth giant samples like the Kandahar giant.
Yeah.
I want some DNA from that. I don't know how I'll get one, but overseas they're not as guarded about it as they are here in the States. I'm hoping I can come up with.
I'd start pestering people in South America because they dig this stuff up pretty regularly, and yeah, they don't seem to have the deep state breathing down their neck down there as much.
If somebody's got connections down there, that would be great because I don't have that many, but I would love to have, the different races of giants represented. We've got 2, but they're going to be the same race. I would love to have the.
What are they? The Anakim, the conehead guys?
Oh, I've got those too, but that's different.
Okay.
They're not really giants. There are some giant ones, but what I've got is regular people-sized ones. No, I'm talking about like the Lovelock Cave giants. I've got 2 of those kind of samples, but I would like to have something like the Kandahar giant, bigger than these.
I know somebody else that was in country there that had another sighting of those things, and they were flying in a helicopter across a valley in the mountains, and apparently the noise from the helicopters kicked up 3 of them, and they took off running across the valley at right angles away from the path of the helicopters. But everybody in the helicopters could look down and see these red-haired giants on the ground running, which they were guesstimating were doing between 60 and 75 miles an hour, by the way.
I'm sure. Look at the foot. They can run, 45 and above themselves.
Yep.
They've not been known to chase people in cars.
Yep. I had 2 different witnesses on my show that have been chasing their cars, and both of them said they had to get up above 50 miles an hour to pull away from them. Until then, they were hanging right with them.
Well, I had a local here that was on a 4-wheeler, and he kept up with them at 45 miles an hour, and he just ran out of woods.
Yeah.
But
yeah, same thing here. As soon as they topped over 50, then it couldn't hang with them anymore. Well, one of them actually helped them because he tried to grab the back of the car, and all he did was give it more traction, so that it really got away from him faster.
Yeah. So, these things can do things that we wouldn't expect they'd be able to do, but they're able to do it. So, which is always interesting. We, touched on that earlier. But, back to the DNA, it's unique. It's not anything that's been seen before. And, this is what's interesting about it. And the fact that they're part human, that they're a hybrid, that's interesting too. I had a whistleblower come forth that I'll release some of that information along with the new study. And they had done some DNA, and it corroborated mine. And they had like 13 bodies in a live capture. And it was very interesting. I've got data that's been given to me from them before they died. And so, I'm not the first. I'm not going to claim first. I'll gladly share it with this other scientist, in memoriam. But it was very interesting. All of the reason that he said they never came out with it was they kept fighting over whether it was homo or not, if it was true homo. And because they tried to make it fit an evolutionary model, and a hybrid doesn't fit an evolutionary model.
Right.
And that's where the big debate came in. So they never came out with it, but I think also the government suppressed that too, because it was a secret study. And but there's no way it wasn't real, because he had data that I didn't have, and I had data he didn't have, and between the 2 of us, it was really interesting. So, and I hate he's gone. He was a wonderful guy. But and I didn't know him. He looked me up. So
the taxonomic designation in his study delineates a reference to a South American reptile, which you would think has nothing to do with them, except for one interesting thing that it can do, which is regenerate lost body parts.
Yes, and that is something interesting that is talked about in this alleged Miller document, how the decedents were regenerating so fast he didn't think they were dead. So that was, kind of corroborated by that alleged scientist. And I do find that very, and about that and other things will come out with the science. I've got all kinds of different things that will be in this. That's why we're not going to try to publish this. We're going to probably put it in the form of a documentary or a informal paper, because the government's not going to let us publish.
No.
Unless I put it out with the same journal as I did that we bought, that we had to buy to get out. And if I do that, it's going to be the same old, oh, well, she self-published. We didn't self-publish, but this would be self-published. Therefore, I don't want to do it that way. I'll just bring it out and let everybody see it when it's done.
As scientists famously said one time, our great spirits have always met violent opposition from mediocre minds. Albert Einstein. And he's absolutely right. Anybody that comes along that breaks their little paradigm and shatters their wall of glass and knocks down their house of cards is not somebody that they embrace and hug and say, welcome, thanks for ruining our world. Now we have to rewrite all our history books. No, that isn't the way you're greeted. So actually, I agree with you. I think that's a better way to put it out, go directly to the people, because look at people like John Bindernagel and how much time he spent and all the study he did, and he wrote books, and he tried to get his, comrades in the scientific community to take a look at it and, actually take a look at it, and they wouldn't. So why waste your time?
Yeah, exactly. I was at the top of my game. And I lost all of my scientific friends pretty much because none of them, either they got threatened by the government or they got threatened by their bosses, or they just were so closed-minded that they didn't want to give it a chance. So, I got ostracized, basically. Now, oddly enough, I had a lot of scientists come forth and contact me and say, man, you did a great paper. This is a great paper. And we believe it, but don't use our name. We don't want to get in the trouble you're in. Don't use our name, but we support you.
We would support you secretly from behind the scenes where it's not helping. Yeah, thanks, guys.
I know we've had a lot of that. And, and our results have been corroborated. So it's not like we're just out there as a lone gun. Some people don't even know they've corroborated them, and they have. So it's really interesting. And all of that's going to come out. I want to finish this thing up. There will be years and years of study. The more money we raise, the more work we can do and the more cryptids we can do. We've got the minimal raise to do, the minimal on all things now. But I would really like to see where we can maybe do some individual genes and maybe some different cryptids if we can get the samples. Like I said earlier, a mermaid's on my bucket list. And, there's other things too, like goat men and other things. We would love to have different cryptid DNA. And because my theory, once again, is that they're all going to have human mothers. I believe that. And I want to prove it. I've already gotten human DNA from the dogman mitochondrial samples, both of them. So I know that they have human mothers.
Well, Bigfoot, just on the face of it, you can look at the creature and you can go, well, I can see how something like that could arise naturally. But Dogman, no. That is not a natural creature. It's fairly obvious just from looking at it.
Sasquatch isn't either. And that's
the point that I'm trying to make, though. On a casual glance, it looks like Bigfoot could be. They're not either. So that really brings into question how many of these cryptids are real and are actually out there, and do they all have the same genesis point, the same mad scientist way back in prehistory playing with his GMO Tinker Toy set?
Well, that's what we're hoping to find out, among other things, because once we get the mito, we can sort of date it. And just like we did with the Sasquatch samples, and they're human especially. And the same sort of with if there's any human in the Y chromosome, which it wasn't with the Sasquatch. They were unique. So their progenitor was not male human at all, not even close. It wasn't anything that has been sequenced before. There's a lot of totally unknown in it. And that's why we're going to revisit even the Sasquatch samples a little bit, because I want to use a different lab and see if we still get the same thing we did with those first 3 genomes. And I bet we will.
Definitely a good idea. Did you get any samples from Eastern Europe by chance? Because there's an interesting thing over there where, in North America, they talk about some of the natives occasionally would have the women kidnapped by the Sasquatch and they would never come back again. Or in a couple of cases, they came back and, they gave birth to weird, twisted things that didn't live very long, fortunately, and that sort of thing. So basically pointing in the direction of they're still mating with human women occasionally. If you go over to Eastern Europe, apparently it's flipped on its head. The Orthodox Church over there had to release an incantation to protect the men from being abducted by female Bigfoot. And apparently this is enough of an issue that the church actually made an incantation against it that they passed around. When Rich Soul was on my show, he actually had dug the whole thing up and read it. But it's specifically for, hey, I'm a good Christian man. Go back to your cave in the forest and be a good Sasquatch. Don't take me with you. I'm not interested in being your husband. And okay, what the hell is that all about? And if there was something like that actually going on, maybe you would see examples of there being male human DNA in the sample.
Rare, let's put it that way. I think the majority of the breeding population, is probably thousands of years old.
Mm-hmm.
But there are some recent ones. But when you compare,
keeping in mind, they made this incantation just a few hundred years ago, so that wasn't thousands of years ago. It was enough of a problem for the church to do something about it.
Right. Okay, let me put this in perspective, though. There are thousands and thousands and thousands of Bigfoot creatures all over the world by different names. I mean, there are I would say well over 1 million if you go worldwide. And they're not uncommon. I mean, the clan I deal with, it's got 10 in it that I've seen at one time. Mm-hmm. So that's not counting babies. So, there's a lot of them. And if they abduct 20 or 30 men, that's a negligible sample. So what I'm saying is the chances, the likelihood of getting a human male sample is highly unlikely, just by the numbers. But it does happen. It's happened in North America.
It's actually happened even more recently in Mongolia in the 1960s. There was a teacher that was kidnapped by 2 Almas, apparently female, and after having them in their cave for 3 or 4 days, they got bored with them and let them escape.
Oh, there you go. Well, like I say, there's several stories, here in North America. And, your co-host, Robin McCrae, when she's on with you, she knows of one too. So, it happens. It's just happens. And when you look at sheer numbers and how hard it is to get the sample, the chances of actually getting one are pretty rare, but I'm sure they exist. Let's put it that way.
Yeah. I just kind of found that interesting because in North America, you get all the old stories about the Sasquatch stealing the women. And then you go over to Eastern Europe and it's all no, the female Sasquatch are trying to kidnap the guys. What the hell,
Yeah, well, they do that. I mean, it's a two-way street.
Apparently those Eastern European guys are really cute because the female Sasquatch like them. So look out if you live over there, guys.
Well, I guess somebody who's looking for a mate should always go over there. But
or perhaps less flatteringly, you guys look a lot like male Sasquatch, and they can't tell the difference. You guys can decide which one it is.
Well, regardless of it, it could work both ways. I will say this, that we have reason to believe that over the millennia, they have genetically I know genetically they've bred out a lot of the human on the nuclear side. We're the weaker species. Right. And, genetically speaking, it's survival of the fittest. So we're hairless. We're not as strong. We're not as tall. We're not as, robust as they are. So those that have more human in them are going to be the ones that die off first because
natural
they can't.
Selection. Yep.
So it's kind of
compete with the big ones.
It's kind of like us with Neanderthal and Denisovan. It's only a very small percentage because they were not like us, and we basically were the survivors of the different hominids from ancient times. And so it's kind of the same with them with their genetics. They don't have near as much human in the nuclear DNA as you would think. But I'm quite sure that that's due to natural selection and die-off due to our weaker genetics. And with that has also made it more difficult for them to mate with us because they are more different than they were from us. Because when they started, obviously, they were half human and half progenitor. And, first generation. And then the next generation, when they started breeding together, say they had some hairless ones born and they froze in the winter.
Mm-hmm.
So down through the millennia, they have become more their own species of hybrid with less traits of the hybrid. That's why you have less of them that really look as human as we are. You're going to find the occasional one that does, though. There's going to be an occasional one that looks very, very human.
Yeah. But again, this would explain why they'll lean over time more and more heavily toward the progenitor male branch of the genome, because the natural selection favors them to be like that. If they're more like us, they tend to die. So
exactly. So that's what I'm saying, and we did find that with the final analysis of the whole genome was that we didn't have as much human in them on the nuclear side as we expected. But yet that mitochondrial DNA is incontrovertible. It's there. So they definitely did come from humans on the mother's side.
Now, that brings me to another question that somebody's going to ask in the chat, so I might as well just go ahead and bring it up right now. Inbreeding, they aren't the only things that hybridize with humans. We've got a good percentage of people from Europe that still carry Neanderthal DNA around with them. Is there any evidence that the Sasquatch have inbred with the Denisovans or the Neanderthal?
No, they predated them. They were gone by the time Sasquatch came along.
Okay.
And we did not find any Neanderthal or Denisovan in our samples. Now, that's not to say that statistically there isn't some, because there could have been, the human mother could have had some in her that passed down, but it's so little that it didn't show up.
Right. So if they hybridized with the human mother that had like 3% Neanderthal DNA, you wouldn't expect it to carry down into the children anyway.
Right. And it will in people, but it wasn't 3% or anything. There was nothing that we could tie it to. We even individually ran the red hair gene, the human red hair gene.
Mm-hmm.
There's one point mutation that is totally Neanderthal red hair. They did not have that. The samples that we did on that individual test for the red hair gene, they had the human red hair gene, but they didn't have the Neanderthal one. So
for folks that think that they might be some kind of a hybrid of us and Neanderthal or us and Denisovan, no, wrong. They're not.
No way. Not at all.
Good to know.
Because I thought they were leftover Neanderthals at first. I really did. That was my first theory.
So did the Russians. They worked on that basis for a long, long time.
Right. And but it turns out that is just not the case. So, with that said, I've just negated everything about Denisovans or Neanderthals to them because there just wasn't any proof that any of it was there. And if it were, it was so little it didn't show up. And if it were, it came from the human side.
Mm-hmm.
So.
Well, that's interesting. Look here. We are narrowing it down. If you can't tell exactly what something is, you can eliminate what it isn't. And we're doing that right now. We're eliminating what it isn't. It isn't Denisovan. It isn't Neanderthal. Thank you for making that very abundantly clear. You guys think it's a Neanderthal? Forget it. It's not.
Yeah, it's definitely not. And I know what they are. It'll come out as far as their genetic makeup. Not what people are going to really expect.
So are they? Does it seem like they're an amalgam? Like some of their DNA is just chunks from other things put together?
Yes, they're genetically engineered, regardless of how it was done or who did it. They definitely are genetically engineered. They definitely couldn't have occurred naturally. That's for sure.
Well, I'll
They
tell
are
you,
not a creature.
yeah, just from observing them and seeing their behavior and stuff, it always made me wonder if there was like some gibbon DNA in them, because man, they got a lot of similarities with those guys too.
Well, you can look at the orang pendek, which is an interesting creature because it has basically an 8 foot. The big toe sticks out to the side like your ape.
Right.
But yet they have the attributes of a Sasquatch in that they do things and aren't easy to find. And actually, Dr. Tom Gilbert from Copenhagen, and on my YouTube channel, I have a little short clip of this. He said he did an orang pendek hair, and it turned out human mitochondrial DNA, just like that. And the people where it was taken, they don't have red-haired people there. And it was bright red hair. And plus, it was apparently a good chain of custody. But he got the same treatment I did. They said, oh, it must be contaminated.
Right.
No, I know him. He is a consummate scientist. There would be no contamination. So it supported my findings with the orang pendek.
Right. Now, the orang pendek, of course, everything over there is orang something, which just means forest person, orangutan, orang pendek, orang moal. There's a whole bunch of them. Anyway, orang pendek is a little short guy for everybody out there that doesn't know. He ain't very tall. And it's interesting that he has the ape-like foot with a divergent hallux on it. And yet, from what I understand, all reports and everything seem to indicate he's a biped.
Yes, he is. Yeah, they are. And that's why I say they're basically another race of something similar to a Bigfoot. And then the DNA showed a human mitochondrial DNA, just like my study did, which was really interesting. Not surprising to me, but interesting. So, you have these creatures all over the world, regardless of whatever moniker they put on them, that they have very similar capabilities. They have very similar, they're bipedal, and, all of that is supports that they're all connected. They're all somewhat the same creature. They are not just a standard ape or anything. They're different, and so regardless of what you call it, they're all going to be similar.
Well, absolutely. And I mean, you'd expect there'd be regional variations. There's one group living in an area for a long time. They're going to, adjust to that area and may look a little bit different than the ones a few hundred miles away, but it's still pretty much the same thing. And what always surprised me is just the observation from the field researchers of like some of the things they do here in North America. And then you talk to somebody in Australia that's looking at the Yowies going, yeah, they do all that here too.
Yeah, and that's the way it is worldwide, regardless of what flavor they are. Mm-hmm. You got skunk ape, you got, and there's some stuff I'll bring out about the different variants and different morphology of even North American Sasquatch. That's something I haven't released yet. So there's a lot of very interesting little tidbits out there about these things and their interactions with people or lack thereof. Some are more gentle than others. There's just all different things to be learned. And I've been so blessed to have, so many people come forward, as well as, my own experiences to learn about this on top of the genetics. And it all fits. It all is very similar in a lot of ways. And I find that fascinating.
Definitely one of the ways you can tell when you're on the right track, when all the puzzle pieces you've got all start fitting together and making a picture, and there aren't any ones left lying around upside down or anything. You can be pretty sure you're on the right track at that point.
Absolutely. And that's what we've got. And I'll tell you something, even the non-genetic field research and other things that I have access to or have done myself, I never take it on one person's word or even my own word. I have to have it verified through an independent source before I consider it part of the data. There's still the science there. I don't, go running out in the woods with cameras. That's no. But if something happens that's an interesting finding of some sorts, regardless of what it is, it needs to be independently verified before I'll even consider it.
Totally agree with you. And you know, somebody's got to be out in the field filming them. I'm one of those guys. Hi. But I might even be worse than you on that score, because I will personally see some weird little tidbit happen, and I want 3 or 4 different people to verify it independently to me before I even start thinking there's a pattern.
Well, let's just go basics here. Everybody knows they make stick structures.
Yep.
It's verified by more than one person, so it's pretty much you would consider it more or less a scientific fact that they make stick structures, because there's too many people that have witnessed them, and there's no explanation for them other than the Sasquatch doing it.
Well, the usual argument there is that whoever saw Bigfoot building a stick structure or tree structure, and my argument to that is, who have you seen building it besides a Bigfoot, and why do we keep finding Bigfoot tracks around tree and stick structures if they're not building?
Well, you know, I was just going basic there, that, you know, there's things that are kind of verifiable about them.
Yeah.
And you want to do that with science. That's why we did a blind study and double blind study in some cases with these samples.
Yeah.
But it's the same with every part of the research. For it to be real research, it needs to be corroborated like our DNA was. And it needs to be corroborated by people that are not connected. So, And I've been very adamant about that when people have told me things or I've experienced something.
Well, you bring up tree structures, and this is great because this is one of those things that's, you know, hands-on, real physical thing. Something is making these tree structures. Is it Bigfoot? Is it somebody else? Whatever, who knows? So you got to investigate it. Now, the next question is, what do they mean? And they do mean something. So then you have to develop a theory on what does this one particular tree structure mean. You have to develop a method of testing if you're correct or not. You have to run a lot of tests and consider yourself wrong or right after a lot of them. And if you consider yourself right, then you start asking other field researchers, hey, have you ever seen this? Check this out and see if it works that way in your area. And a good example of this is like leaners on trees. We started noticing that they would leave leaners on. What are the leaners for? The leaners are trail markers. There's certain trail markers they make that are like secret trail markers. They'll snap a branch. Why'd they snap the branch? Well, because they're going through there. This is one of their regular travel routes. Is the branch completely off? No. Is it twisted? Yes. Which direction is it twisted? Is it twisted to the right? There's a secret trail to the right. Is it twisted to the left? There's a secret trail to the left. This is stuff that we've tested in the field, and it comes up every single time. Yeah, there's something over there. So that's how you start figuring this stuff out. And unless you're doing a lot of it and you've got other people elsewhere doing a lot of it in their research areas and comparing notes, you never figure it out.
I've had things like that happen. That one of the first things that happened when I was introduced to him is I had a pecan tree, and they were breaking the top of the branches out of the pecan tree, and it made me mad. And I was storming about that. And the day after I was storming about it, there was a little bone X at the foot of the tree. I mean, and it was not put there by an animal. There was no way. They were perfectly matched bones, perfectly sized. I have pictures. And it was right there under that tree, and they never broke another branch out of it. So, you know, and other people have stuff like that. And that's
Were they somehow unaware that was your tree? Because it sounds like you had to explain that to them.
It was my tree, and it was my pecan. I was like, breaking the top out of the tree. It's right in the front yard. It's making it ugly. And it's going to hurt my nut crop. So, you know, it was definitely, you know, it was definitely an interesting observation that I was not expecting the next morning.
But then they quit messing with the tree.
Absolutely. Never was it touched.
Yeah, I'd like to make the point on this note, since you just brought that up, and this comes up a lot. You know, if you're having issues with your local Sasquatch and they're like maybe doing something in your property you don't like or something, the first thing you can do is go outside, especially when you think they might be around, but just in case there's somebody watching, and just tell them out loud, hey, you guys quit doing this. I don't like it. Quit it.
Well, I was storming. I was mad. I mean, there was like 3 or 4 big branches out of the top of it.
Don't wreck my tree. It's yummy. Quit it.
So they moved to a bigger tree, and they would go up the back side of it, and they wore the bark off the back side of it, chewing the tree. So I think, they're watching is what it is. They watch.
Favorite observation tree.
Exactly. So, I mean, it's been interesting. They did not come out to me until I did the study, or started the study. And after I'd actually seen them somewhere else. But it's been quite the trip. And, I've got lots of pictures, gifts they've left, all kinds of crazy things. But, that's not science, so I don't usually talk about it. But I'm not the only one that experiences this. So the point I'm making is it's been corroborated by others. Things like that happen.
Yep.
So,
that's the point I'm making, too. Some of these people that have been doing field research for 10, 20, 30, 40, longer, like me, and a few of my friends. We've seen so much of this stuff in the field. We've been talking to witnesses that have had weird things like this happening to them for like decades. And it gets to the point where they can start telling you a story and you go, and then this happened next. And they're like, how'd you know? Because I've heard this story 100 times.
A lot of times.
Yeah. Or my favorite one is, I got Bigfoot messing around on my property, I think. I'm not sure. But there's like something slapping my house and it knocked my wood pile over. And I'm like, let's see, it's springtime right now. They're moving around. Do you have a drainage ditch or a little creek or something near your house? Yeah, there's one about. They're following the drainage ditch around and they're messing with your house because it's right next to it. Don't worry about it.
Oh, I tell you what, and some of the things they've left me have been just unbelievable.
Yeah, some things they consider to be really cool and a gift are like not on our gift list.
There's been dead birds. There's been live crawfish that I've never seen on property before.
How the heck does a live crawfish get up on your porch?
I'm sorry. Oh yeah, right in the front door. Right at the front door, no doubt.
Yeah.
And I had one thing that a big rock. I wasn't sure if it was rock or if it was plaster that had imprint of a fern in it. Looked like a fossil possibly, and I was going to chip on it and see what it was because they had brought it up and left it by the house. And I was talking to another person, a friend of mine was over, and I said, hey, this thing is, I can't tell if it's plastic or if it's actually kind of a fossil. I said, it's pretty neat though, isn't it? Well, the next morning I go out and it was broken in half to show that it was not rock. It was broken in half plastic. I don't know where they got it. I don't know, but they left it. So
some of the interesting things. My friend Joyce Gray was out walking around filming them randomly. And she had this one that used to show up pretty regularly. And one of the times she filmed him, she sees him over in this bush and she's getting ready to zoom in on him. And he sticks his tongue out at her and holds up his cell phone that he stole from somebody. Like, what the hell, dude? And then there's another guy over in Michigan who was walking around his research area and he's filming just kind of, he's going out into this field, and on the edge of the field, there's, of course, the wood line, and ways up the wood line, there's something standing next to this tree, and it's too close to him. So he doesn't want to cause conflict, so he just kind of like brings the camera across and then he keeps going to get out of there. And when he goes back and looks at him, there's a Sasquatch standing there, and he's got somebody's game cam, and it's got the batteries ejected, and he's holding it up like it's a trophy or something, like he's proud of it. And it wasn't one of this guy's game cams. He has no idea where it came from. But he went back to check the spot, and sure enough, there was nothing standing. There wasn't any Bigfoot or anything standing there. So
I've had a National Honor Society medal left in the seat of my truck. I don't know where but they did. And it wasn't mine, because mine were in my jewelry box inside. So it was somebody else's. And it wasn't shaped like any of mine, but it was a National Honor Society medal. They've left costume jewelry in the catfish. You name it, it's been weird, some of the things they've left. But You know, it just
Oh,
goes with the territory. Once they're around, they're around.
yeah. My main camp out site up there in the Sapphire Mountains, they had started leaving weird things on the campsite, and I didn't realize what was going on. And one time we show up, and there's a pair of brand new Oakley sunglasses and really expensive motorcycle gloves, and they're just sitting down by the river. Like, who the hell would leave those there? And then the next time we show up, there's a brand new canteen, all shiny silver, beautiful. Who leaves this stuff around, you know? So I got curious, and I asked Robin, where is this stuff coming from? She's like, oh, they think maybe you or your guys lost it when you were walking around in the woods, so they're bringing them back to the campsite in case you lost them. Then you can get them back. And I asked everybody, and they said, no, we didn't lose that. So I let her, tell them that, we didn't lose any of that stuff. She said, they said, oh, good, we'll keep them then. I don't know what they're doing with Oakley sunglasses or a silver canteen. But the thing that occurred to me immediately is that there's other campsites in that valley. So where exactly are these objects coming from? We lost them somewhere else in the valley, like where? On somebody else's campsite?
Oh, boy. Yeah, I mean, it's crazy stuff. I mean, I had trouble with them turning the water on and leaving it on.
Oh, that could get expensive.
It'd get hot in the summer, and they'd turn on the water, and they'd leave it on, and I threw a fit. They hadn't done it since, but I threw a fit. I was raging about that. And one night, a juvenile opened my front door and twisted the doorknob so hard that it busted it. I had to take a chisel and everything else to get the thing out to replace it. It was so bent up. And that was something that a normal human really couldn't do. And the same night, they turned on the water, and brand new brass faucet I had on that, and they bent that to where I had to replace it.
Oh, God.
So, yeah, I said, stop bending things.
Did Robin show you the picture of the dog kennel that's got the drop hasp that goes on to the big post that holds it in place? And she was complaining that the dog kept getting out. So they caught the dog and put the dog in there, and then they crushed the hasp. So, like, you literally couldn't open it anymore. They had to replace it. It was just destroyed. It's not opening.
Like, they don't know their own strength. I mean,
no, they pinched it together. That's not how you use it. You don't drop it in place and then pinch it together. You just drop it in place.
The only way to keep it on, I guess. But, it's been a wild ride since and like I say, they came out to me not too long after we were still doing this study. I guess they came out to me about 2 years before the study published. And, it's just been a wild ride ever since. Like I say, I love them. They're great. They're kind and thoughtful and everything, but they're good neighbors. Let's put it that way. But they do some really crazy things.
Yep. And if you're friends with them, they're some of the best friends you can have. So I'll tell you that much. It's worth being friends with them.
Not mean or bad in any way. They're amusing, actually. They've done some. One of these days, if I get brave enough, I'm going to take all my pictures and make a book out of it with what happened along with it, because it's some pretty amazing stuff. Like the raw crawfish wanting to crawl in my house. That was. I know what crawfish, where their little homes are, and you can tell it. And I've never seen one on this property. Yet I've had 2 different live crawfish, one at my front door and one at my front gate. And actually, I saw another one the other day, and there are no signs that they've ever lived here. So it's been a wild ride. What can I say?
It's interesting how they import things. They decided when Kevin was down in Arkansas that his backyard was too wide open, because even though there were trees there, they had knocked down all the underbrush and they mowed it. So the light penetrated from the back porch too far into the backyard, and they couldn't effectively hide and watch him sitting out on the porch smoking a cigarette when he came out at night. So one of them decided to rectify the situation by moving a tree into place where he could hide behind the tree, and then Kevin wouldn't see him. The only problem with this is that 35-foot tree suddenly appearing in the backyard was pretty freaking obvious. It still had the roots on it, and it had clay on the roots from there's no clay within 5 miles of his house. So apparently they ripped this thing out of the ground and carried it quite a ways to have just the perfect shelter to hide behind when they're watching Kevin smoking his cigarettes. So yeah, they do really goofy stuff sometimes.
Yeah, that tree that they've got the bark rubbed off of, it's over 100 years old, and like I say, they go up the backside of it, and they go way up in the top and sit in the fork. And I mean, you can see exactly where they've been and where their hands have been because that bark is rubbed off. Mm-hmm. So yeah, they watch, and that's what they were doing in that pecan tree they were killing, and it did finally die even after all. So the kids play on the roof and go over. I have a peach tree, and they would go pick the peaches off the top of on my roof. They'd run across, get up there and pick the peaches and sit up there and eat them and throw the pits down and the partially eaten peaches. So, it's they do a lot of things. That are very interesting.
Yeah, they're very humorous and they're fun to hang around with. And my favorite thing that I can do is go out in the woods. I know most people are like, "Ooh, gonna go camping where Bigfoot is, spooky. I'm scared." And with me, it's just the opposite because I've been camping with them for so long. I know they're not going to hurt me. I totally trust them. I feel safer up there camping with them than I do sitting in town at my apartment.
Well, they're safe to be around. I used to go out in the woods at night all the time and get around them. I mean, seriously, I did that for years.
Yeah.
But then I had a dogman encounter, and there's other things out there, and I don't go out in the woods anymore. I'm not going out in the woods anymore because that thing was violent. It killed a dog and hurt a horse, and not going there anymore.
Sounds like bad dog man. My both the groups that I hang around with seem to have dogmen hanging around with them because I've got them on video in both places, and both times they're with the Sasquatch.
The Foots left whenever the dogman showed up, and we're gone for a few weeks.
Yeah. Yeah, up here they seem to get along. That one group that's been up there for a long, long time, they've got at least 2 dogmen that are hanging around with them because they got 2 different ones on video with them now. And both times there was like big groups of Sasquatch with one lone dogman with them.
I wonder if the dogmen that hang out with the Sasquatch aren't really a Sasquatch with a little bit of dog DNA thrown in them, possibly, so that they're more Sasquatch than they are anything. Whereas the ones that look more like just upright walking wolves, I'm thinking that those are the bad ones. really bad.
The one I got last fall that had 3 Bigfoot around it, not to mention all the other ones that were in that video clip. He looked like a black German Shepherd. And the one that I got the previous year that was standing on his hind legs peeking around a tree and had a Sasquatch on each side of him, he was kind of brown and looked like a German Shepherd. There was nothing ape-like about either one of them. They could have passed for an average dog.
More the body maybe more shaped like a person.
Yeah.
No, I mean, it's just a theory that may or may not hold water because I haven't really talked to anybody about it. I haven't really studied the dogmen like I did the Sasquatch at all. But they kind of, since they've been on my plate, since I've got the DNA, their interest is now there.
Yeah.
And the fact that
I personally think there's more than one thing going on with the dogman. There's more than one thing that are being called dogman that are running around on the planet.
There's got to be some explanations for it. That monk that hundreds of years ago wrote the more or less scientific dissertation about the city of dogmen. It was very interesting, and it seemed like that they were fierce, but they weren't like constantly killing anybody or anything. It was they had a city, and anybody that's interested in dogman needs to go on YouTube and find that video and watch it because it's very interesting what this monk wrote about them. And the same with the Marco Polo, like you mentioned, he also came across the city of dogmen.
Yep. And the other thing is there's multiple reports of them from the ancient period up to like the medieval period. And besides them being, stated to be, well, if you can actually hire them, they make a great mercenary army. The other thing is that they basically said they weren't very violent. They kept to themselves. They couldn't speak English. They had like barks and growls for a language or whatever. And they were great traders. They would trade with humans. You could trade things with them.
Well, the Foots traded with the Native American peoples.
Mm-hmm.
Now, there's a lot of oral legend there about them trading with. In fact, in my books, I broached that actually, that they were trading with one of the characters who was a Native American woman. And so, it's interesting, all of these historical references. And you can learn a lot by studying history prior to all the science studies and everything, and even the field studies.
Yep.
I highly encourage that. There's I don't know if it's still up. There used to be a website called the Very Hairy Dictionary. And it had over 100 different photographs of Sasquatch. And in Europe, like they're on cathedrals even. Yep. There are pictures of wild men on cathedrals. There are tapestries. There is art. In the Sistine Chapel, there's even a drawing that has Sasquatch in it in the Sistine Chapel.
We've showed it on my show before too. Yeah, it's right next to a river. There's like a little underground cave, and one side of it there's a male and a female Sasquatch, and the other side there's hell.
Yeah, exactly. I have actually seen that. I was in the Sistine Chapel. It's there.
Yep.
And so it definitely there's so much history out of Europe, which is older, of course, than the U.S. And they're all the art. So you're going to see all of that. But once again, it goes back thousands of years before that, too.
Yep. Yeah, I had Robert Boston, Bigfoot in Germany, on my show a few times, and we've done, Rich Soul and I did a couple of shows on European Bigfoot and whatnot, and there's just no doubt they're all over the place. When Robert was over there, he was getting a lot of pictures of that kind of local stuff. And, one of the little towns he lived nearby, right in the middle of the town square, they've got the water fountain. Every old-fashioned little town had that, right? Oh. So what's the water fountain? It's a statue of a Bigfoot. Yeah. They're Wildermann.
Yeah, that's what I'm saying. They're everywhere. And, and they are hybrids. They are human hybrids. The mothers were all human, of course. They do not have DNA that matches on the paternal side. There's huge blocks of unknown DNA that doesn't even align with, even partially with anything. And like I say, most of the DNA has been done on most species on Earth now, or at least, the mammals are pretty well done. And with that said, normally, in fact, you can look this up online, is they'll say that chimpanzee is almost human, Actually, that's a kind of a misnomer in that they have the same genes we have, about 98% of the same genes we have, but their actual sequences are quite different. The sequences.
Aha, yes. As a matter of fact, dolphins have more sequences similar to us than chimpanzees do. And bananas have 97% the same DNA too.
Well, that's what I'm saying. There's kind of some misunderstanding there. We're not like chimpanzees.
Yeah, no kidding.
But
this is what they always use to justify that and say, oh, we're the next door relatives to chimpanzees. Bullshit.
Yeah, we just have the same genes pretty much.
Yeah.
But they're completely different sequences. And the sequence is what makes a person, or an animal, or whatever. It's gonna determine your physical makeup, your internal makeup, your, everything in you is determined by your sequence. And then there's, the genes are just, each gene has a function, but they're going to be different sequences in different species, very different.
Right.
And
well, that's what I'm saying. By that line of logic, then we're more closely related to dolphins because they've got more of the same gene sequences we've got than chimpanzees too. So that's fallacious logic.
But they also have, it's kind of a neat little chart where it shows you the percentage of gene alignment. Mm-hmm. Like the chimpanzees, way up there in the high 90s, whereas bear is like 80% and going down to dog and chicken and all these other things about how the same genes in people are 30% in a dog or something. So, you can look at that and kind of get an idea of things, which is interesting, but the sequences are totally different. Every species is very unique to themselves, even if their genes align. So, and the simpler the species, the more alignment with sequence there is. But by and large, there's so much misleading things out there that people that don't know science, they don't understand it. And they make assumptions based on what would make sense to them. I mean, I see where they misunderstand it. But we are not chimpanzees, and they are not us. And
we aren't dolphins or bananas either, as far as that goes.
Exactly. Interesting thing, though, chimpanzees, their chromosome 2 is inverted, which is a big difference. You have to have a karyotype to really, work with that. But they have a inverted leg on the chromosome, arm on the chromosome. So there's some interesting things out there genetically that between species, and I'm going to address some of that too in the new study. There's going to be a lot of stuff come out that people aren't expecting with this new study, because it's not going to just totally be just like the last one wasn't total genetics. It had the hair, it had the electron microscopy, it had other things besides the genetics. Well, this is going to be similar, but it's going to be a little different. And I think people are going to be really excited to read and understand, or watch and understand what's going on. And I hope we can raise even more money, because the more money we raise, the more we can reveal, because we'll be able to do some testing as well as other things to bring forth a more balanced picture of what they are and how they are, and other cryptids and the same type of thing. I mean, there's some real interesting things I have not shared that I will share. So I'm hoping people will donate to the Cryptid Genome Project, we're calling it. We have a GoFundMe, because the more we raise, the more we're able to do testing and what have you, and put this all together so that everybody will eventually have all this knowledge.
And we will have the link to her GoFundMe listed in the notes section on the video. Might even be running in the scroller on the screen right now. Go pay attention. Throw wads of money at her Throw all the money you can afford to burn at her, then dig up some more and throw it at her. Understand? It is your duty. Go forth, fund Melba Ketchum. She is putting her butt on the line to actually do this research. She's put her butt on the line before, and they kicked her butt. But she came back, and she's going to keep going, and they can't stop her. So, being that Americans love underdogs and people that tell the truth, let's throw all the money that we can afford to throw at Melba Ketchum and help her with this, because it's the very least we can do. The very least we could do is help fund this epic project.
Well, I appreciate it, Duke. I mean, like I say, we're putting the money toward these extra samples we have, and any that we will get in, and that's why I'm not just putting it. We set a goal for the minimal, but the more we raise, the more we can do, and the more cryptids we get from people. So I'm putting out a plea also. Anybody that's got weird cryptid stuff, it's not just Sasquatch. We would love to test it. And, it'll be no charge to you as long as we're raising research funding. So,
Well, I'd send you my peanut butter container with the big finger marks in it, but I'll guarantee that's Sasquatch, so you probably don't want
yeah. We've kind of got that under control, although we are going to do, 2 or 3 more with a different lab just to confirm our genomes a second time. But, there's just so much out there, but we can kind of kick this all together in one nice little tidy package, I think, if we can, raise enough to test everything and tie it together. And then we'll know what all these things are. We'll know at least their genetic makeups, and that will give us a lot of information. And then, we're not going to put any of the money toward field study or anything. Although some of that will be involved in this. I've talked more about it with this interview than I ever have. But, there's a lot of people doing, field stuff. They're out in the field. So
yeah, we got that covered. Don't worry about it. Just tell us what kind of sample.
I'm not worried about it. But there'll be this kind
what kind of cryptid do you want a blood sample from? We'll get one. Just tell us.
I don't want blood. I want hair with roots. And any kind of weirdness, actually,
How about some skeletal pieces?
I want some marine biologists to come up with some merman or mermaid DNA. I want that. Just for grins. A thunderbird, you mentioned a thunderbird. I'd love to have one. That'd be interesting.
Anybody who lives over in the Philippines, shoot a rope and send her some samples of that. That should be an easy one to prove.
Well, like I say, I think they're going to be related. I may be wrong. We'll find out if we get, more samples to test. Like I say, what we got now is basically, like I say, dogman giant, elongated skull, and a couple of unknowns that seem to be hominid. And we got Xena and Quit, but I'm not, like I say, they're at the bottom of the stack because there's already been some DNA done, and this would be just to confirm what was already done. Good scientists did it, so I'm fairly trusting of it, and I would like to, in the future, possibly do it, but that's only if we get enough to test everything we're going to test.
Don't scientists use the Reagan axiom that he used against the Russians when they said, "What do you mean you want to come and check our nuclear stockpiles?" And he said, "Trust but verify." Oh, we trust you, but we're going to verify it.
I like to verify everything. I mean, ours has been corroborated. Even unknowingly, it's been corroborated. And I'll bring that out, too. Both by a whistleblower that was dying as well as others. So, I'm and like I said on the first part of this, is we had DNA sequence for the mitochondrial DNA that was human from other labs before. 3 other labs actually had found human for Sasquatch samples. And then we got the samples and did the nuclear, and it was unknown and different. So we knew right away that these things were not human per se, not real human, pure human or anything. So we took the same samples, we got the same mitochondrial sequence, and then we turned around and got nuclear sequence that made them different from the same samples. So we proved that they are not just human samples, which is what a lot of people say, or they think human contamination. And once again, I have that video on my YouTube channel about contamination. We have proof they were not contaminated by any other mammals. we used universal primers, and it would have been an overlay of the two sequences. So they can go watch that to show that that was just a put up thing.
That's one of the dodging disinformation tactics that the pseudoscientists have been using for a long time. Oh, we tested the sample, but you must have touched it, and because it came back human. Well, don't you know how to test a sample?
Well, Dr. Tom Gilbert is a very good scientist. He's the one that did the Thana first round testing. And like I say, I would just corroborate his probably. That's why it's at the bottom of the heap.
Yeah.
But he also did the meringue Pendec, and it was human on the human mitochondrial DNA. And he was attacked just like I was. And his lab is a consummate lab. He published tons of papers scientifically. And they called him, oh, they must have sweated on it. Your lab people must have sweated on it. Seriously, he got so mad he won't mess with it now. And it's a shame. We've lost a good resource there of a good scientist that was willing to look at this stuff. And now he won't, because he got so angry because the documentary made him to look like an idiot, just like they're trying to do me. Mm-hmm. So, it's not just me, it's anybody who touches it. And that's why I had so many scientists come and say, you can't tell anybody that I'm supporting you, but that was great work. They don't want anything to do with it because they'll get attacked. And it's a shame, because it's something that's valuable that needs to be done, because these cryptids are out there. They exist.
Yeah, just because somebody's giving Dr. Ketchum hell doesn't mean that they're reputable or reliable. There was recently a book that came out that the jabroni spent about 10 years compiling it just to attack Dr. Ketchum. And he had some other actual supposed scientists write a foreword to it. And apparently it was so researched and edited that they missed the fact that they misspelled the word forward. Don't listen to people like that.
Well, I'll tell you a little something. The psych study within 6 months was discredited by Cambridge. If my study was so horrible or so contaminated or so messed up, I guarantee you there's a line of scientists standing in line to discredit it.
Yep.
I would have been wiped off of the planet by naysayers with genetic proof that I screwed up. Or not I, but our team screwed up.
Yeah. Well, where is it? It's been how many years now? Where is it?
Yeah, the only people I know that have tried to do anything are not even geneticists. Or they don't work in the field or never have worked in the field of genetics. There's been PhDs that have tried to come against it, but they're not qualified to even review it, much less come against it. So, show me the...
Does it tell?
Oh, my goodness. Yeah. Well, he came... I'll tell you where he discredited himself is right when it published, he immediately came out with, just raging against it. And then the guy that was interviewing him asked him, and it was actually Joe Rogan, I believe. I may be wrong. It could have been the Houston Chronicle, but it was one of them that came up and said, asked him if he had read the paper. And he hadn't even read the paper, but he was raging. Now, that's real scientific. If you don't even read the science and you're sitting there saying it's not good science, it's got to be contaminated.
You might find this interesting. One of my friends who used to be on my field research team was actually going to college to get a bachelor's in anthropology. And for the topic of his final dissertation to get his bachelor's, he decided to pick, could Bigfoot be real? And he extensively cited your study and also some other basic facts about distribution and food resources in North America. And he had like everybody in his class convinced Bigfoot 100% exists, and he got an A+ on it. So there you go.
Well, like I say, anytime you get into science that is not already proven and it's novel, and another quote that Disotol did is he said that we used technology so new that he didn't even know about it, or he didn't do it.
So, like, that's your fault that he's still got training wheels on his bicycle? No, that's his fault.
And I know that was, I believe, on Joe Rogan too, or it was, I believe that, yeah, that one was on Joe Rogan for sure. So y'all can go back and pull that archive and listen to it. It's really amazing. We did. Dr. David Swinson, who is a consummate scientist, he was the first person to map the Staph aureus genome. And so he has extensive, and he about blew his computer up, checking out our sequences and stuff. And he corroborated what we did early on. And he's also on Joe Rogan. Actually, that clip is on my YouTube channel where, he was not a believer, but, he got the same thing we did, basically. So,
Yeah, the guy you keep quoting is catching these guys in an interview isn't a believer either. I'm starting to like this Joe Rogan person, however.
yeah, I kind of agree, but, I was invited on, but he was hostile and I just wasn't going to deal with it. Why should I have to? Let somebody else try to disprove what we've done. Our team did a great job. I mean, I did not write that paper. I wrote portions of the paper and I put it all together, but each person that had the expertise wrote their part of the paper, as it should be.
Right.
Like, and that did the whole genome sequencing, he wrote that part of it. The hair analyst wrote that part of it. The person that did the electron microscopy, he wrote that part of it. Because, nobody has all of those expertises. So we had each individual, and they were all highly valuable scientists. We had an MD on there that did the histopathologist. He was a pathologist. He did the histopath. So we had experts in every discipline in the paper, and it was a multidisciplinary paper, write their own part of the paper, and then we just put it all together. And I put it all together. And, and I wrote part of it. I wrote part that, some of the parts that our lab did or we outsourced, and I handled the data. So it was a real team effort. But the day that it came out, they had so much blowback that they were calling me frantic because they were going to lose their jobs to feed their families and everything. And since I was the first author, I took fall. I let them go and said, just refer everybody to me. And that's how that started. And they think that they sound like, oh, me, the kitty cat vet, has done the whole paper. That is far from true. We used 12 labs. And, our part was very minimal as far as anything in that paper because, our one little lab, everybody would have laughed, oh, one lab has these results, ha, ha, ha. That's why we sent out all these samples to different labs. We didn't tell them what they were testing. And so we got a really nice, cross-section of the genetics of Sasquatch. And so, it's done with the greatest of scientific care and with wonderful people that were just the best. Like my second author, he actually has written papers on DNA extraction. And he's got state-of-the-art tools to do it with. And, so, he wrote the protocol for the Tomb of the Unknown Soldier, the one they did where they identified I believe, Vietnam era Tomb of the Unknown. Mm-hmm. So, these people are not just, sliced bread. They're the top of the field. The one that analyzed the electron microscopy was head of the department at Texas A&M. We had it was UT that did the University of Texas, did the whole genomes, and that was who wrote that part of it. All of these things we checked, we double checked, we did everything in our power to put as much data in there. It was so long that we had to put a lot of things like materials and methods in the actual supplements rather than where they normally are in the paper because it was so long. Mm-hmm. And that was courtesy of the Nature editor. He said, this is too long a paper. You have too much data, so we're going to have to put some of this in supplements. So one of the things that he suggested I do is put the materials and methods in the supplements, which I did. And the guy that didn't read the paper comes back, where's your materials and methods? I said, they're in the supplements. The editor told me to put them.
Derp. Oh, my God.
It's just, ridiculous. But we did good science. And I will stand behind my co-authors. One of them called me in a frantic that somebody had called the lab and said, how can you have a scientist working for you that believes in Bigfoot? That's not good. That's part of what ended my career is they'd get me in court and they would ask me, do you believe in Bigfoot? Well, I'm obviously not going to lie under oath. Well, yeah, I do. And then they would laugh. And make the jury look at me like I was a crazy. And so that ruined our forensics forever.
Well, the correct answer to that one is yes, I already proved that they're real. Aren't you paying attention?
Well, I did. I would go on and say that we wrote a scientific paper, you know, proving their existence. But, you know, it didn't matter because all the time, defense or whoever was on the other side would be giggling and, you know, making a show. So, you know, but that's okay. I mean, actually, I enjoy writing, and that's what I do now a lot. It's, you know, I write books. And so that's what I'm doing.
Well, I enjoy bludgeoning trolls with a mace, and I do that on pretty much every day. So trolls that plan on showing up on this show, you fell for the trap.
Well, you know, the trolls are not fun, but, you know, it's just.
Oh, no, they're fun. We like to embarrass them, then ban them. Please show up, leave stupid comments so I can embarrass you, then ban you.
Well,
that's what's going to happen.
I'm sure you'll have plenty with me because I tend to have a big red target on me. But
well, I'll be interested to see because the majority of the trolls on my channel, we pretty much annihilated. There hardly ever any new ones show up anymore. I have probably like 500 people banned from my channel right now.
I'm sure that people will, you know, I mean, it's like anything that can discredit me because of my knowledge base and because of what I've done, you know, they're out there doing it. And,
oh, we look forward to it. The more of the actual shill trolls out there that we can get banned off my channel, the better. So bring it on, trolls. Looking forward to banning you. And in the meantime, Melba has the link in the show here. Make sure that if you have extra ching-ching and you want to find out the scientific truth about these cryptids, please donate to her study. She is going to do spectacular work just like she did last time. And where do people find you online?
On Facebook, on Twitter. You know, basically that's my main two. Facebook's probably easiest. I have a public page, Dr. Melba Ketchum, and then I have my private page, but it's pretty well filled up. I pretty well got close to 5,000 people on it. So I only have about 50 slots available for more friend requests.
There you go.
But, you know, Twitter always, but I don't post as much on Twitter as I do Facebook. I'm really not a big poster. I mean, I'm a very reluctant person to be in the public. I'm very private. I don't post much about my personal life. I don't post much about anything other than science. And sometimes I even go a while without even doing that. Because I'm, like I say, I'm just a human being like everybody else. I'm nothing special. And I really don't like being singled out as special or anything or having fans because to me, we're all the same. I just have an expertise that I was trained to do. And that doesn't make me better than anybody else. You
can't avoid it. You were destined. You know, your last name starts with a K. Your special K. Get used to it.
I understand that that's an ex-married name. I'm really not even that.
Okay, so Dr. Special K is when you find out more stuff in the future. Let me give me an assurance that you will be so nice as to actually come back on the show again and update everybody.
Oh, I'll be glad to. Yeah.
Awesome.
I don't do interviews very much. I don't like to. They need to be affiliated with radio because there's hundreds of podcasts out there, and I just can't do them all. So I limit it, and plus thank
God I don't have a podcast. Whew, boy, that's a relief. I don't do podcasts. I've never been on a podcast network. Actually, I had my own radio show for about a year, but I quit doing it. You know why? Because you can't show the evidence on a radio show. I want to show the evidence. That's why I do this show.
Well, regardless of it, it's, you know, I'm very careful, and I really don't like doing them because it always just stirs up the haters, and, you know.
Yeah.
And then I get a bunch of emails that I don't have time to answer, and that's embarrassing.
I get to deal with that every day, too.
Everybody, I try to at least answer everybody. And if I get a whole flood of them, like I get every time I do an interview, I miss a few, and then I feel really bad because I really do try to personally answer everybody. And, you know, at this point, it's getting unmanageable. So...
Same thing with me, guys. If you send me a message and you don't hear from me right away, I'm sorry, but I'm only one person. I'm doing the best I can to try and do everything. And when you're cranking out a show every week, it tends to pile up. There's a lot of comments, messages, people need help with things, et cetera, et cetera, et cetera. If you don't hear back from me right away, well, send me another message. If you pester me enough, I'll probably get back to you sooner. That's about the only thing I can promise. I do the maximum amount that I can, being one person with limited amount of time to do all this stuff. But again,
Ditto.
what
Ditto
I,
here.
thank you so much for being on the show. This has been one of the things that I always wanted to do since I've been doing Bigfoot shows was I want to interview Melba Ketchum because I adore Melba Ketchum and all the work she's done for the not only the Bigfoot community, but the Bigfoot, which, hey, some of them are my friends. So I really appreciate that. Just get the word out there. Hey, these aren't monsters. Quit shooting them, you idiot.
Well, now there are some that are, though.
So
You
there's a
can't
lot of
say
humans that are monsters
there
too, and I bet there's a bigger percentage of human monsters than there are Sasquatch monsters.
Oh, I can guarantee that's true. I can guarantee that one. Having worked in forensics with all the monsters that come through the penal system, oh yeah, people are worse.
Oh, the ones that are running the governments, those are the biggest monsters.
Well, yeah, I'm just a scientist. I don't talk politics. I have them, but I don't talk them.
I do. I got a whole conspiracy channel, and I'm a conspiracy analyst and a geopolitics expert. So I will roast their butts. And if you missed the most recent one where we totally roasted their butts on a Beyond InfoWars level scale, go over to my Rumble channel and watch History of the Knights Templar Part 2, Death of the Knights Templar, Rise of the Banksters, to get what I'm talking about.
Well, there you go. I'll let him do it. I'll stay neutral.
You're already in enough trouble. At this point, we just don't even care anymore. We're just going to attack everything that's evil, period.
I just want to have a peaceful life. That's all I want. Stay on the farm and have peace.
Yeah. I just want to live out in the woods with lots of Sasquatch neighbors and have everybody else leave me alone.
Yeah, that's about where I want to be. So we're on the same page there. So anyway, I appreciate you having me. It's been a really interview, and I've way deviated from the science, and I hope that people will forgive me for that. But
well, no, that's okay, dear, and I think people are going to really appreciate it, and it's going to be refreshing because a lot of the stuff that you did in the past strictly focused on the science. And so it's more interesting, especially for my audience, because believe it or not, 85% of my audience probably have either seen a Bigfoot or had a Bigfoot experience. It's a very high percentage. So they are totally on board with what you're doing, and they're willing to give you 100% of their attention to listen to what you're saying, unlike some of these other audiences you might get stuck with.
Well, partially also, I've done so much of the science, and it's repetitive because I haven't come out with the new stuff yet, the new data. And so it's kind of boring to rehash them every time. So years later, here we are with actual interaction, and things are different, but it's still the same science.
Yeah,
Science supports it. I think the future studies will too, but it'll just widen our understanding.
New cryptid data. Oh boy.
That's much stuff I haven't shared yet. And then I've got new cryptid stuff. So.
Yeah, and see, I've gotten wind of that too, that there's some stuff you didn't share yet that you've already got. So, if you want to come back and do a part 2 or anything, just let me know whenever you want to. I'd be more than happy to have you back again.
I'm going to pull all of this together and do one big reveal, and then I'll probably do a bunch of interviews.
There you go.
But we've got so much. I mean, it's like when we wrote the paper, we only did some limited sequencing on the whole genomes for the paper. Now we have the whole genomes analyzed. We had a bioinformaticist that analyzed the entire genomes. We have nice little pie charts showing what's in them. And I have not released any of that.
Ooh, juicy.
Yeah, there's going to be a lot of stuff coming out that nobody's going to see coming.
Awesome.
And it's not boring to talk about that there's neat stuff going to be coming out. And, and I'm talking strictly, pardon me being really lax with my scientific jargon tonight, calling it stuff instead of data, but I know the difference. but
It's my fault. I dumb all my audience and guests down so that everybody can understand what they're saying.
there's going to be so much coming out that nobody's going to see coming, and it's going to be so much fun to reveal it. So, and we're not going to test though until the fundraising pretty well ends, because we want to see how much we're going to get, because we'll have to allocate the various monies to do what we need to do. And so we don't know what we're going to all be able to do except the bare minimals right now. We've got that covered, and that's great. But the more we raise, the more we can do.
Right.
And once we get to where it levels out and there's really no more donations coming in of any size, then we're going to figure it all out and see what all we can do and amaze y'all even more. And that's the goal.
I am just ridiculously excited to hear what you come up with. And I know I've already heard some of the leaks from the stuff that you haven't leaked yet.
Oh, who did it?
And I'm not saying nothing until you decide you want to come out and say something.
It's going to get updated. They got to sign an NDA, because I don't want it leaked like the project was leaked.
Yeah.
And I will go after them this time.
And that's, and I always agree with that. NDA, no damn aliens. Never do any dealing with aliens.
Well, that would be another interesting DNA sample, would be from an alien.
Yeah. And if that shows any human DNA, there's something really wrong going on.
Not necessarily, because what I understand, and I don't study this, mind you, but what I understand from people in the know, some of them are hybrids. That they do collect, when they abduct people, they collect their genetics, and they do use it to create creatures and people-looking ones and what have you. So I did have one that looked human that swears they were a part alien contact me, and I was like, "Get away! I don't want to talk to you. I don't want to see you." She wanted to meet me. I'm like, "No, no, 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 no. We're not doing that." I don't want to be part of those. But, there's a lot of, and a lot of it's coming out and being corroborated. What's interesting is it's actually being corroborated by people that are ex-CIA whistleblowers at this point. And you'll do this on your conspiracy theory. There's a guy out there that talks about all the underground bases, the DUMBs, and he knows what each of them was for and where they're at as far as any underground war. And there's 3 locations in North America where they make human hybrids. And one of them is Montauk, by the way. Mm-hmm. So it's very interesting. And they do, even out in the open now, we have the technology to do gene editing with CRISPR, and so you can actually make these things now.
And that's how they make crispy critters with that CRISPR technology. And we did a Conspiracy Corner episode on Phil Schneider, the first guy that talked about the DUMBs. And even at the time that he was talking about him, he was saying there was more than 30 North America. And I know somebody that claims that they were in one under Michigan where they've got a zoo collection of cryptids that they have stashed away down there.
Oh, I don't doubt it a bit. This guy, he had a map, and I mean, this is kind of off subject, but it's interesting. And he went through each of them and said what their purpose was and whether or not the so-called white hats or the black hats were still controlling. And back last year, and then a few years ago, we had these unexplained explosions around here. And it scared everybody. They were calling the sheriff's office and everything else. I don't scare easy, so it didn't really bother me, but it was interesting. And they all came from a certain direction, not too terribly far from where I live. And, one of them was big enough it rattled the windows even. And they'd come at night usually. Well, fast forward to last year when this alleged underground war is going on, we had one great, huge explosion that was the same direction, same location as these previous ones. I think they were building it and then they blew it up. Just my humble opinion. And what was interesting is when this guy came on with this map of these DUMBs, there was one right where these sounds came from, which I found interesting. So who knows? But
There might be something to it. I saw a video about a year ago where a guy was talking about that, and he had video of there's a city, I'm not going to name it, underneath it. Apparently something was going on because there was fire coming up out of the sewer grates.
oh yeah, I know all about that. Go for it. It was very interesting. And the other thing is this guy overlaid earthquake over his map of the domes, and they corresponded.
Yeah.
And we actually had an earthquake right around the time of that big boom, and we don't get very many.
According to Phil Schneider, they were using boring technology, big machines to make the tunnels in, and then they deflagrate a huge cavern using small nukes. So if they were making one, that would make the shock wave. That would make you think it was an earthquake or something because they're making one. But on the other hand, if there was already one there and somebody decided to get rid of it and collapsed it, that would also make a big shock wave.
Well, they say they're using tactical nukes to get rid of them. That's, what this guy was saying. Anyway.
Yep, that would do it.
Yep.
That would definitely do it.
It's not genetics, so I don't need to be talking about it. It's all conspiracy.
Yeah.
Just interesting stuff I like to read. I mean, my Achilles heel is my curiosity.
Yep. All that UFO thing, that's not hard science. We don't have any evidence for them. Bigfoot, on the other hand, we have DNA. Take that, UFO people.
I would love to check it against the unknown factor.
Send Dr. Ketchum some samples of whatever it is. Make sure to include picture, date, sample taken, and location where it was retrieved.
And preferably a type of sample that can be washed, like hair with a root or teeth or something like that. Although, if it's an interesting enough sample, we would do saliva or blood or something like that. But we prefer not, because those things will contain bacteria. Even if they're a clean sample for mammals, they will contain bacteria. So we're trying to keep it just as clean as possible.
Yep.
But the bacteria is not going to affect the outcome of the data. It's just going to be there. So.
Right on. Okay, any last words before we go?
Thank you for having me.
You're very welcome. I've only been hoping for years that I would get you on the show, and I'm so very happy to have you here. And like I said, open invitation to come back and give us updates. I know by the time this show airs, you folks will already probably be aware of the fact that she was on Spaced Out Radio earlier this week. And we might even try and set her up with an interview on another big radio show too. So anyway, keep the attention, keep eyes on Dr. Ketchum and help her, because she needs all of our help. And meanwhile, you guys all know the drill. Don't go getting lost in the woods. Don't pick on people and be mean. And don't feed donuts to the Loch Ness Monster. And the main and most important thing ever, do not hug the Wookiees.
Oh, boy. I love it.
Everyone, safety first, last, and always. Pay it forward. Don't be mean to people if you don't have to be. Don't flip off the mountain giant. Don't poke Dogman with a stick. Don't punt the puckwudgie. And for God's sake, whatever you do, do not hug the Wookiee.